Yesterday, I talked about chloroplast gene, PSAC gene, knockout mutant by chloroplast transformation. I hope you still remember. Uh, PSAC gene encode iron sulfur protein of photosystem 1 complex, and when we knocked out this gene, we knew already that this gene encode iron sulfur subunit. But we already knew that this gene is essential for photosystem 1 function. So that why we knock out this gene is because we didn't know what happened to the PS1 complex in the absence of PSAC gene product. For whether the partial reaction of PS1 is still assembled, functional, or the whole structure of whole complex is assembled, accumulated, or not accumulated, or destabilized. So we want to know some additional information than the function of this gene product to be an acceptor of photosystem complex. So this experiment was kind of forward genetics. That means if you have genetic information and gene information, and from the homology of other genes, this gene encodes some function. You can expect phosphatase or kinase or some other enzyme activity. You can speculate. Okay, so this is forward genetics. But I'm going to talk about today it's reverse genetics. So usually forward genetics is you know the phenotype of the mutant and you find out the gene that has a mutation. From the phenotype you can identify the gene of the mutation. This is normal way, okay? Then you assign the clone the genes, something like that. So this is one of these things. But reverse genetics is if you find some open reading frame that you don't know if it is transcribed, translated, that is expressed. Of course, we don't know the function at all. So then we try to knock out this open reading frame, then to see some phenotype. If we see phenotype of this open reading frame, must be a gene encoding some important function, functional protein. So this is reverse genetics. We generally call this genetics. So I think I just remind you of the transcript of PSAC gene. This is this is southern. Okay. Northern analysis I described in the Y type. There are two messenger RNA which are hybridized with PSAC gene probe. And smaller one just transcribed of only PSAC gene from this size. Not more than one gene is transcribed. But there was another larger messenger RNA, 1.1 kilo base. And we mapped the 5' end and C' 3' end of messenger RNA. So we found that the small and large messenger RNA are transcribed at the same position, but the larger one, end of messenger RNA is a little bit longer. So we suspect there must be some gene, although this may be small, this must be small, but there must be some gene or open reading frame. Okay, then we sequenced this region. Okay, at that time we didn't have whole genome sequence of Chlamydomonas. Okay, so today's title is YCF7 encodes a small subunit of cytochrome B6F complex that is named PET-L. And forward genetics, PSHC gene knockout is forward genetics versus reverse genetics today. Okay. PET, the gene name, this is protein name, and lowercase p, PET-L is the gene name. The photosynthetic P stands for photosynthetic, E electron transfer, electron T transfer. So PET genes encode a protein that are involved in electron transfer reaction. So this is one of the subunit of cytochrome B6F. So this is named PET. And according to the order of the discovery, is alphabet is added. So PET-L is. So PET-L is this small subunit is gene is PSHC, and in the downstream YCF7. Now here it's written in the PET-L. You may not see, but this is YCF7, hypothetical chloroplast open leading flame 7. Okay. Then after we assign this gene product as a small subunit of cytochrome B6F, it was renamed as PET-L. Okay. So we sequenced and then found this is the downstream region. And downstream of PSA gene, we found the two open reading frame. Open reading frame 58 and YCF7. This is also open reading frame, about 30 amino acids, very small open reading frame. But this sequence was conserved in other chloroplast genome and even in cyanobacteria. Okay. So that's why, but this open reading frame 58 was found only in Chlamydomonas. So we suspect that this open reading frame may not be functional or transcribed or expressed. Actually, we knocked out this open reading frame and we didn't see any phenotype. So I'm focused on this YCF7 and how we assign this protein open reading frame as a B6F a complex component. This is a predicted amino acid sequence of YCF7. So it's very much conserved. Some organism has a longer. These are 2-methionine, but we don't know which is initiation methionine. So it's described like this. It's very much, and this is a hydrophobic region mostly. So this hydrophobic region forms alpha helix one time. So this is a single transmembrane. This contains single transmembrane helix. Very small. So we knock out opening frame 58 by inserting AD cassette and also YC7. As you can see here, the physical map and the restriction map. Open frame 58 has unique restriction site, DRA3. And YCF7 has a unique restriction site, SNAB1. So we inserted, respectively, AD cassette at DRA3 or SNAB1. And automatically we can, how to say, when we clone this, to make this transforming vector, at the same time we can insert AD, we can get two types of the transforming vector in which AD cassette was inserted in the same orientation, no transcription orientation is the same as this PSAC to YCF3, the same direction, or opposite direction. And this is also, we could make both. So we transformed 4 different, transformed chlamydomonas, chloroplast with these 4 different transforming vector, okay, respectively from each. So we could get 4 independent transformers, okay, and this is a Southern blotting, and total DNA was digested with restriction enzyme EcoR1 and the PST1. PST1 here, EcoR1 here, and PST1 also has here, 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 and here. So you can distinguish this different the AD cassette insertion at a different orientation by the size of the Southern blotting signal. You see, the different orientation, the position of PST1 is different. So this is a result. They're very similar to band are migrated here. So you may see only single band. It's actually very closely migrating to band. So DNA is fragment, and this is completely different. This one, okay. And the position of this is probe with the probe this region, and also AD cassette region was here lower, and this upper. This contains AD cassette, and here, and also here. This one. This one is very. This one has almost no YCS3, so this one, and PSD. So probe did not react very well with this because of the position of the probe we used. We couldn't detect, but we obtained 4 different transformers. Then we tested the photosynthetic growth on agar plate with the medium, HSM medium, that is a condition for photosynthetic growth. Only photosynthetic growth can possible here. And this is types of control, heterotrophic condition, photoheterotrophic condition, and this is photoautotrophic condition. So this is control. This is sorry. First row is Y type control strain, and okay, these two Y type control, Y type control. This is PS1 minus mutant as a photosynthesis deficient mutant. This is negative control, so it didn't grow like this. Okay, and second row ORF opening frame ORF 58 grow well. So we didn't see any phenotype related to photosynthesis. So we just stopped thinking about this. Okay. The third row and fourth row are YCF4 knockout mutant. Okay. The same and the opposite orientation of AD cassette, so didn't change a lot. So it grow heterotrophically, but didn't grow photosynthetically very well. It slightly grow, but more slowly. So photosynthetic activity was rather affected. Oh, so this YCF7 product must be somehow involved in photosynthesis. Okay?
So what was the third one where the phenotype is missing?
Which one? This one? This one is here it grows heterotrophically, but this didn't grow autotrophically. Because this is PS1 deficient mutant. PS1 deficient. It's not B6F, but it's PS1. So this is a negative control. How it grows, PS1 minus photosynthesis minus mutant, how it grows. Okay. At this point, of course, I already told in the beginning that conclusion. But when we were working with this, we didn't know YCF7, what YCF7 encode. Right? Okay. So we found that this is somehow related to photosynthesis, but not essential, important, but not essential. Okay. And we also, we raised the antibody against YCF7 product. So this opening frame was overexpressed in the E. coli, and poor rabbit was injected proteins, no? Overexpressed protein, and then generated antibody. So we nicely made antibody, okay? Wild type and control. Well, we couldn't detect any signal in YCF7 mutant. So this also confirmed that we have successfully generated YCF7 knockout chloroplast transformant. Okay? And also it shows that YCF7 is translated. YCF7 is a gene, encoding a protein, very small protein. So there are two things observed. Then we want to know where these proteins involved in photosynthesis reaction. So this is an interesting. It's a little bit small, but if you look at this panel A, this is immunoblocking, okay? And this T is total cell protein. So we just solubilized the whole cell, and solubilized whole cell protein was migrated, separated on SDS polyacrylamide gel electrophoresis and detected. For example, we used cytochrome F antibody and YCF7 antibody to know where this YCF7 is localized, which compartment. Of course, we think that it must be in the chloroplast, but this is, and also this is a hydrophobic protein, so this must be present in the membrane fraction. So to confirm this, we separate after breaking the cell, and then we spin down, and then pellet is the membrane fraction, and the supernatant is soluble fraction. So we separated pellet, P, and S, supernatant fraction. So pellet is membrane proteins recovered, and S is soluble protein recovered. And then this is a cytochrome F represent the membrane protein. So membrane fraction P gives us a good signal, the same signal as a total cell protein, but no signal in soluble fraction. So, okay, so we could separate perfectly the membrane fraction and the soluble fraction. And YCF7 was recovered in pellet fraction. So, as expected, YCF7 is membrane protein. And we also purified from the membrane fraction, because membrane fraction contains many membranes, many different types of membrane. Cytoplasmic membrane or other organelle membrane. So we try to purify more by sucrose gradient of the thyroid membrane. TK is the thyroid membrane fraction. Then cytochrome F is separated, localized here. And YCF7 was also present in the thyroid membrane. Okay, so, okay, we conclude that the YCF7 is thyroid membrane protein. Okay, so next is panel B. The chlamydogram has many photosynthetic mutants deficient in a component of out of many PS1 photosynthetic proteins. So, for example, we tested, because this is thyroid membrane, so we tested Y type and FUD7. This is photosystem 2 deficient mutant. Okay, and FUD6 is cytochrome B6F mutant, deficient mutant, and F15 also cytochrome B6F mutant, deficient mutant. F15 is photosystem 1 deficient mutant. Okay. So then, as you can see, the cytochrome F is PET-A is component of cytochrome B6F, is present in Y type, PS2 deficient mutant, and the PS1 deficient mutant, but lacks deficient in B6F deficient mutant, as expected. And YCF7 is, as you can see clearly, the pattern is the same as cytochrome F. It's present in the Y type, PS2 minus mutant, PS1 minus mutant, but deficient in cytochrome B6F mutant. So, okay. In the absence of B6F, the B6F deficient mutant, probably B6F has many subunits, but the mutant mean mostly all of the component are, in most cases, it's gone. Okay. So, okay, so then we can conclude YCF7 is a component of cytochrome B6F from this mutant analysis. And you think it's enough to confirm that this is a component of B6F? So we didn't think so. Then we thought, if we purify cytochrome B6F complex biochemically, YCF7 should be present in the purified cytochrome B6F preparation. And we did this experiment. And actually, this is a collaboration with EBPC, Institute of Biology Physico-Chimie in Paris. This is French, okay? Institute of Physico-Chemistry, okay. Institute of Biological Physical Chemistry Institute. So there is one researcher that is Dr. Popo. He's engaged in for the first crystallization of cytochrome B6F. He was trying to crystallize a B6F complex from Chlamydomonas at that time. No. I think I show you B6F complex data before. No, not really. The swinging pH dependent photosynthetic control. Okay, but anyway, he told us that in his very pure preparation, he didn't detect YCF7 polypeptide. So he claimed that, oh, there should be the YCF7 should not be present in our preparation. Okay. But I went to that institute and then purified the B6F and then checked. Cytochrome F, the purified B6F preparation shows, and also YCF7 gives us very strong signal. Okay, so there was, but the purified preparation, if you separate by SDS polyacrylamide gel electrophoresis and the stained protein, actually we couldn't detect the stained polypeptide because we found that this polypeptide was so small and was not stained by Comasy brilliant blue or something like that. Only immunoblotting we could detect. So you know that sometimes immunoblotting is a very powerful method to detect the signal. Okay. So we found in the B6F complex this protein is we confirmed it's a component. So both data, mutant data and biochemical data confirmed this is a component of B6F complex. So we named this PET-L. Okay. So we have to name the gene after the confirming the function or expression and function. So what this doing, this cement is what doing what? The question arises. So YCF7, that is now PET-L mutant, did grow autotrophically, but at a slower rate compared to a wild type. Why? So we carefully analyzed the accumulation level of cytochrome B6F in YCF7 mutant and wild type. We loaded the same amount of chlorophyll, but wild type 100%, 75, 50, 10. Okay, this is a dilution series to estimate the quantity of the polypeptide in the mutant. So if you look at this cytochrome F, you see that is signal intensity is less than 75%, more than 50%. So it decreased a little bit. Okay. And here also the other component is also the same, between 75, 50%. Okay. So it's a little bit decreased. But the growth was rather significantly reduced. Okay. And we isolated before was the complex isolated before I showed before was B6F from wild type. So we also isolated B6F complex from wild type and YCF7 deficient mutant, knockout mutant, and compared this subunit. So the wild type and the mutant, W wild type, M mutant, cytochrome B6, cytochrome F, cytochrome B6, subunit 4, iron sulfur class center. This is non-specific cross-reaction with this antibody, but this YCF7. The other small subunit, PET-G, PET-M. So after purification, the signal intensity is almost similar, except for, of course, YCF7 is missing, and FES protein, that is the PET-C proteins, are lost. This was present in the thyroid membrane here, but after purification, this was dropped off from the preparation after purification. So this means that this is absent from the beginning, but this was lost during purification. The others are relatively well maintained. So we speculate what happened to this mutant. Do you think, I hope you speculate a little bit, why this phenotype happens? What do you think? You have any idea to explain this data? Sometimes the data is, we have to speculate to explain. We have to explain, by thinking, speculating what this data means. Okay? Any suggestion or any? Although in the thyroid membrane or cell, FES subunit were normally present, but after purification, this was lost. Probably solubilization and the subsequent purification process, this subunit was dropped off. Why? What happened to the B6F complex? So this is an interesting point of the research. Okay. Data, how to interpret the data. That is unexpected, sometimes as expected or unexpected. As expected, it's easy to explain, but unexpected result, it's not easy to explain, but this is the most interesting point of research. Is there any suggestion? Or this is very important. What do you think? No suggestion? Anything? Okay. Our interpretation is binding of this FES subunit, PET-C subunit to the B6F complex was destabilized in the absence of YCF7 subunit. Not so difficult interpretation. So, and then we can conclude YCF7 is important for the stabilization of cytochrome B6F complex. This is conclusion, our conclusion. So the function of this is something like that. Then if that is the case, we think what we can do, what is the next plan to experiment? Which type of experiment we can do? Okay, this is also important. This is only speculation, okay? But we need to evidence to support this, our conclusion. Huh? Oh. Sorry, I missed the data to include. Sorry. Then I talk to you. So we thought if this without YCF7, B6F is a little unstable. So the cells growing logarithmically, logarithmic phase. It's the protein is actively synthesized. Okay. So accumulation level of B6F must be very closer to wild type level. But if the growth reaches to stationary phase and or growth phase is a stationary phase, cell didn't divide, so protein synthesis is most of protein synthesis is terminated, stopped. Okay, so the most protein remain, but unstable protein degrade, decreases. So we tested accumulation of cytochrome B6F complex in the cells from the logarithmic phase, late logarithmic phase, and stationary phase, and compared the accumulation. And then we found that in the logarithmic phase, B6F was rather, depend on the subunit, it's because of sensitivity of between sometimes 75%, okay? But in the stationary phase, the level of accumulation was very much decreased and then very low, 10% or. It has happened. So even in vivo, not during a purification, the B6F complex was unstable. So this is the balance, steady state level of in the logarithmic phase cells. This accumulation level is the equilibrium between synthesis and degradation. So synthesis is high in the logarithmic phase. Accumulation becomes high. But once protein synthesis activity decreased in the stationary phase, accumulation level starts to decline. That's happened. So this YCF7, PET-L, small subunit, are very important for stabilize the B6F complex. Although this is not essential for the activity of B6F complex. So that was our conclusion. Sorry for that. This data was I missed to include. So this is a reverse genetics. So if you have unknown genes in the genome, and if you want to know, you can do reverse genetics. But if you do randomly, this is not fruitful, wise method, because the function of the gene is not focused on only chloroplast photosynthesis. But this small gene was present in the chloroplast genome, So there was a high possibility that this small open frame is related to photosynthesis. But if other genes in the, for example, nuclear genome, and very interesting, the gene that of which function is known, but if there is a unknown genes or open frame next of this, and then if co-transcribed, you can expect some relation, right? So you can do reverse genetics in that case. Okay. The last lecture of the following lecture, I will talk about another topics of side-directed mutagenesis to introduce point mutation into the chloroplast gene. So I will stop my talk. So today and yesterday I talked about knockout of chloroplast gene. One is gene we know already the function. So that is a forward, similar to forward genetics. And today's talk is unknown, just putative, open frame. So hypothetical chloroplast open frame of which function is totally unknown. So this is reverse genetics. Okay. So we, from the open frame, we reach to the function. Okay. But usually, forward genetics is you isolate a mutant with phenotype of your interest. Okay, then you try to identify the gene of the mutation. Okay, to which genes involved in responsible for the phenotype you are interested. So this is a forward genetics. Okay, so now I ask you questions, comments. Is that okay? In the chloroplast, as you may notice that PSHC and YCS7 genes are co-transcribed. But these two are related to photosynthesis. But one is photosystem 1, the other is B6F complex. So they are not encoding the component of the same complex. So we don't know why this co-transcription occurs, but its related function or physiology, we don't know yet. So, and other chloroplast genome of a vascular plant, this arrangement is not conserved. Okay, I think you understand fully, you understand, or totally difficult to understand. Okay? Or is there anything that you couldn't understand? Which is the point that you couldn't understand? If you let me know, that's helpful for me. Is that all right? Okay. If not, I finish my talk a little bit early, but if I put too much, so if there is some, could you close this? How to close this? Here. Escape. And then if I do this, okay. Yeah. I lost this. Where did you? I lost.
It was here.
Here.
I studied.
Okay, it's nice.
From University, Vizag. Sir did PhD from School of Life Sciences in University of Hyderabad only. and he did postdoc at CPMB, Usmania University. And after his postdoc, he joined as assistant professor in GIAN University. Sir has expertise in plant physiology, proteomics, and genomics. Okay, sorry, we have talk after tea. Yeah, you join after tea.
Hi, good morning to all. I hope you are enjoying the workshop. I think Venkat already introduced about me. Thank you, Venkat, for your kind introduction about me and my work. So I would like to congratulate these organizing members of this workshop. Yes, yeah. The organizing members of this workshop, and I would like to congratulate you also, so patiently listening to talks from morning to evening and actively participating in the sessions, tutorials, as well as all these things. And today I'm going to talk a very small topic, I think related to a very important component, call them as a phenomena, call them as a acclimation. So what do you mean by acclimation? What do you mean by acclimation? Suppose you travel from very distance to University of Hyderabad. Yes? The moment once you reach here, I think you feel some discomfortness. Yes or no? But after 10 days and we are about to finish this workshop, now you feel comfortable. Yes or no? Yes? Yeah. So means you have adapted. Means we have adapted or we have adjusted the specific environment. A specific environment. So this phenomena is always called it as a acclimation. Okay? Acclimation. Then here the word is called as photo acclimation. What do you mean by photo? Photo means light. Okay? So because light is the major source of energy, and according to the first law of thermodynamics, in the process of photosynthesis, the light energy is converting into chemical energy. Light energy is converting into chemical energy. But the amount of energy which is present in the universe is equal. Okay, so but the energy can be converting into one energy form into the another energy form. So this is called as that is energy neither be created nor be destroyed, but it can be converting into one form of energy into the another form of energy. So this is called as a first law of thermodynamics. Now, so I would like to ask one important question here, because I'm going to talk this concept regarding about this climate change. I think everybody is talking about climate change. So I would like to ask one question here. Is climate change real or not? Our climate is changing? I would say no. What is your answer? So how do you say that our climate is changing very rapidly? Only flora and fauna? Why flora and fauna is changing? This is the science of climate change. So here, I think intense drought episodes, we are experiencing heat waves, rising sea levels, melting glaciers, warming oceans. So these all are the different signs of climate change. Then what are the factors which are driving the climate change? The most important factors which are driving the climate change is the emissions of the unprecedented increase in the greenhouse gas emissions. I think you all know that, I think in the form of modernization, in the form of civilization. So the human beings are moving from these sustainable practices into the unsustainable practices. So when we are trying to make more unsustainable practices, so we are releasing more greenhouse gases into the atmosphere. So the moment the threshold levels of greenhouse gases are increasing, so which create lot of problems to society as well as our environment. So I think you all know that. I think so nowadays I think everybody is facing a lot of health complications like bronchitis, cancers, different types of diseases and all this. Because of all these are most likely to be integrated with the climate changes. Integrated with the climate changes. Now, see, I think you just see this. I think this figure. I think among all greenhouse gases, the carbon dioxide contributes major proportion. I think see here, I think around nearly 76 proportion, followed by the methane, followed by the nitrous oxide, as well as followed by the again the fluorochlorocarbon gases, here. So these all are emitting into the atmosphere, and which are causing hell of problems to the human individual as well as our climate, as well as the environment. Now, another question is, does CO2 rise follows temperature rise? I think you just see here. I think this graph clearly indicates that. I think these are the CO2 levels before the industrial revolution or modern human modernization or civilization. I think there were, I think you know very well, the CO2 concentration used to always, I think they are measured in the form of ppm or micromole. What do you mean by ppm? Parts per million. Parts per million. Okay. Now, see here, before industrial revolution, I think what's happening here, I think in the year 1880, I think, so what's happening here, I think the CO2 levels were around 270 parts per million, but due to modernization and civilization, so the CO2 levels are continuously rising, and now, I think in the year 2019, it got reached around nearly 410, now the CO2 levels would be around 420 ppm. So you just imagine that in a span of nearly 100 years, I think there was a significant rise in CO2 levels, So I think you must have feel very hot nowadays because of this rise in CO2 levels. Now, see here, at present, I think the CO2 level would, I think is around 420 ppm. Again, so if you won't take any preventive measurement, if you are not conscious, what's happening here? I think by the year 2050, I think it would be reaching around nearly 550, and by the 2100, it would be reaching nearly around 700 ppm here. So you just imagine that. The moment once it reached to CO2 levels would be reaching around nearly the 700 ppm. I think there must be rise in average global temperature by means of this. I think 3 degrees centigrade here. So you just imagine the severity of the climate change. So when I was working at University of Hyderabad, so the CO2 levels would be around I think 400 and around 395 or 380. Now you just imagine in a matter of I think 10 years, it got changed nearly. I think it is reached nearly around 420 ppm. So you just imagine that every year there would be rise in nearly 2 micromole of CO2 is increasing. Without, I think, so this is the clear symptoms regarding about how we are going to face a lot of complications of the climate change. Now, so how to mitigate this increasing atmospheric CO2? So what could be the major root cause of CO2 rising? First one is deforestation. Yes or no? Then second one is extensive usage of fossil fuels. So if you reversing these two processes, definitely we would be able to normalize the CO2 levels. Yes or no? Yeah. So how do you normalize the CO2 levels? So because we have to practice afforestation. What do you mean by afforestation here? Creation of artificial forest. So the artificial forests are also called as agroforestries. Agroforestries are the short rotation forestries. So what are short rotation forestries here? Suppose by you find very huge area which is not using for farming, or you find some degraded land which is not fertile at all. So you just select that area. That area can be used to cultivate the fast growing tree species, and this tree species which will go over a period of time, and you just harvest the biomass, and this biomass can be used to generate bioenergy, and in the form of bioethanol or whatever it may be here. So this ethanol can be acts as a fuel. Okay, so we are partially replacing the fossil fuels. We are partially replacing the fossil fuels by using this biofuels, by using this biofuel. So for this purpose, we need more amount of biomass, So that's why I think we need to efficiently utilize this. We need to efficiently utilize this, the unutilized lands or unfarming land. So that we could able to continuously generate the biomass, and this biomass can be used to produce the different types of energies here, including bioethanol here. The bioenergy, that is the energy which could be generated through, I think this component, call them as a carbon neutral. Why? Because so the amount of CO2 which is released into the atmosphere, again, which will be taken up by these agroforestry here. You just imagine here. So here, these are the short rotation forestry. We just cut down the biomass. And so what's happening here? So this biomass can be used to generate the bioenergy. Now this bioenergy, again, it will release the carbon dioxide. No, again, this carbon dioxide will be sequestered by the same tree species again. So this process will continue over the 40 to 50 years. Now what could be here? So this type of bioenergy is always called as a carbon neutral bioenergy. Why do you call it carbon neutral bioenergy here? The amount of CO2 which is releasing is equals to the amount of sequestration here. So this is called as a carbon neutral bioenergy here. Now, I think this is a well-known phenomena, well-known biochemical reactions. I think you have been seeing from the day one from this workshop. Yes or no? So I think this is the photosynthesis. I think the basis of life on Earth. Without this photosynthesis, there is no life on Earth. Yes or no? So which is mostly comprises by two different biochemical processes. The one is a light reaction and another one is dark reaction. So what is the use of light reactions here? I think the light reactions are always meant to produce ATP molecule as well as NADPH molecule, because this can be used by this dark reactions, Calvin-Benson-Basham cycle, so to sequester the carbon dioxide here. So what are the end products of the Calvin cycle here? Either sucrose or starch molecule. Yes or no? So here again, so the Calvin cycle always comprises two different biochemical process. One is RUBP carboxylation as well as RUBP regeneration. So RUBP carboxylation can be catalyzed by the most important enzyme called them as Rubisco, the most abundant protein. Then again, the another topic called them as RUBP regeneration. RUBP regeneration, I think, so which can be catalyzed by so many biochemical reactions, so many enzymes. But among those, I think simple three major regulatory enzymes are there. What are they? Chloroplast, pseudoeptulose 1,7 bisphosphatase, fructose 1,6 bisphosphatase, and phosphoribullokinase, and 3 are the major regulatory enzyme. Why I call as a regulatory enzyme here? Because these enzymes will always control the flux of carbon dioxide into the Calvin cycle. So that's why I think these enzymes are always called them as a metabolic valves, which will control the flow of carbon into the pathway, so and which could eventually affect this the growth of the different consequences here. Now, so why do we focus more on this Calvin cycle is because people try to manipulate the enzymes of Calvin cycle and the proteins of this light harvesting, that is light reaction, so that people could observe some enhancement in the photosynthetic efficiency, which eventually resulting into increase in crop productivity here. So that's why I think we mostly likely focusing on this photosynthesis, especially I think light reactions and dark reactions here. So in order to perform efficient photosynthesis, I think these two processes needs to be coordinated with each other. So without light reaction, there is no ATP, NADP. Without NADP, ATP molecule, there would be no proper activity of Calvin cycle enzyme. So that's why for a better photosynthetic efficiency, these two processes needs to be coordinated with each other, and without coordination, I think there would be no proper photosynthesis here. Now here, so how to suppose you have 4 different plants with you. Okay, plant A, plant B, plant C, plant D. Okay, so you wish to know which plant is performing better photosynthesis. So in order to track the photosynthetic changes, we have to track light reactions, we have to track the changes in dark reaction, especially Calvin cycle. So how to track this light reactions, how to track the changes in Calvin cycle, dark reaction? So I think you have seen this instrument, I think in SAR's lab, I think you have seen this instrument, that is handy P. So the handy P will always majorly focus the changes in the efficiency of photosystem 2, that is the flow of electrons from the photosystem 2 to photosystem 1. So which majorly emphasizes, I think, whenever we are using the handy P, we could able to see the 4 different parameters. I think Fe by Fm, call them as the maximum quantum yield, followed by the performance index, electron transport, and dissipation. So these all will always emphasize majorly, I think, the photochemical quenching as well as non-photochemical quenching. Later we will discuss. Okay. Then I think you also see this instrument, IRGA. Yes or no? What do you mean by IRGA here? Infrared gas analyzer. So this instrument will always be able to emphasize the changes in the Kelvin cycle, efficiency here. So by using this IRGA, I think we could able to quantify changes in photosynthetic efficiency, water use efficiency, transpiration rates, stomatal conductance, as well as the intercellular CO2 concentration. So what do you mean by what is intercellular CO2 concentration here? I think you know very well. The moment the CO2 will enter into the plant cell, so which will concentrate inside the chloroplast. Yes or no? So I think suppose there is a what I told here, 420 ppm CO2 is there in the atmosphere. The moment once it passes through the stomata, so what would be here? So the intercellular CO2 concentration would be around, I think, 3/4 of the atmospheric CO2 concentration here. So we are seeing now 420. So I see intercellular CO2 could be around 300 or 340 like that here. Yes or no? So here this could be a way, I think, how to track this changes in the, I think, photosystem to, I think, photo efficiency here. Now, by using this IRGA again, we can also track, assess the in vivo carboxylation efficiency of Rubisco. By performing a simple experiment known as A by Ci curves. See here, what is happening here? Here, photosynthetic rates, and here the CO2 concentration. So with that instrument, what you do that, if you provide the artificial CO2 to the plant by program, in this program, we will increase the CO2 concentration from 0 to 1000. So what is happening here? Suppose we just fix a program in such a way that, so the program runs for 30 minutes. Okay. So what's happening that here, we just keep the leaf and hold it inside the IRGA and keep it for that with tripod, whatever it may be here. And so we will fix the program in such a way that the CO2 levels are increasing gradually from 0, 100, 200, 400, 600, 800, 1000. Okay. So the moment once we keep on gradually increasing the CO2 concentration, so what's happening here? So there is a significant rise in photosynthetic efficiency at certain level. After that, I think there would be the appearance of saturation here. So here, now, this typical ideal, which one regarding about this A by Ci curves can be divided 3 different regions. One is which one? I think this is this region will always emphasize this Rubisco efficiency, RUBP carboxylation efficiency. So up to this 400 here, up to this 400 CO2 levels here. Then again up to 700, I think this region will always emphasize the efficiency of RUBP regeneration. Again, next one followed by the triose phosphate utilization here. So what is happening here? Suppose if you rise in amplitude of this graph at this region, which indicates that there is an increase in activity of Rubisco. Suppose if there is a rise in amplitude of the graph around RUBP, which indicates that there is an increase in RUBP regeneration capacity. So this will clearly emphasize that, I think, what's happening that here. So which will analyze this, which will we will come to know that, so which plant is more efficient? Which plant is more efficient by performing this simple A by Ci curves here. I told you, see here, I think Rubisco carboxyl efficiency can be controlled by 3 different enzymes. What are they? Rubisco activase, and carbonic anhydrase. Again, next one, RUBP regeneration can be controlled by, I think, different types of enzymes. What are they? Pseudoheptulose 1,7 bisphosphatase, fructose 1,6 bisphosphatase, and phosphoribullokinase. So these are the 3 different enzymes which could always be able to contribute for the efficient RUBP regeneration here. Now, this is another type of measurement we could use always to track this photorespiration. So how do you track the photorespiration here? I think so here, this is called as a, I think, CO2 assimilation rates versus increasing photon flux density. So what is happening here? Similar with the A by Ci curves. So there was, in the A by Ci curves, we was gradually increasing the CO2 levels. But here in A by Ci, A by Q curves, we are gradually increasing the light intensity. So by using this one, we could able to quantify 3 different parameters. First one is saturated photosynthetic rates, then apparent quantum yield, then dark respiration. So these all are the three major important parameters, so by which we could always assess the efficiency of photosynthesis in different types of crops and different types of circumstances here. Different circumstances. I hope you got this point. Yes or no? Yeah. Which one? Which one? This one? So which one? So at what light intensity the plants are getting saturated? PAR, yeah. Yeah, this one. So suppose you just see the two different graphs here. So one graph here is saturating at lower intensity and lower light levels, and another graph is showing the saturating at higher light intensity. So which one is performing better here? This black one is performing better as compared to this one. Yes or no? So here, this how I think we could always be able to perform the different things here. So people try to investigate the crop responses under changing climate. So people find, I think, the moment when we start exposing the plants toward this, simulated levels of carbon dioxide around 2050. What I told here, by the year 2050, I think we are anticipating the CO2 levels could be around 550 parts per million. Yes or no? So people start doing this, anticipating. People start simulating this CO2 levels by different concentration. So I think the moments when people expose to high CO2 levels, they suddenly observed that the significant rise in photosynthetic efficiency around 50 to 60%. 50 to 60%. Then they also observed diminished somatal conductance. Then they also observed this significantly rise in water use efficiency. Then they also observed regarding about this increasing biomass yield. I told very clearly the changes in biomass or the changes in crop productivity is directly proportional to the photosynthetic efficiency. Photosynthetic efficiency. So more photosynthetic efficiency, more biomass, more crop productivity. Yes or no? So here, then again next one. So the augmented photosynthetic nitrogen use efficiency. So these all are the different ways I think plant could able to perform the different consequences here. So only positive response. They don't observe any negative response. See here, they also observe some different negative responses. I think you must have seen that our forefathers, our grandfathers are more healthy as compared to us. Yes or no? Because they were eating very quality food. So we are moving towards this modernization of civilization. We are not having a quality food. Rather, we are having some different types of foods. Yes or no? So, which are causing a lot of serious health complication because the changing climate is always induces this nutrient dilution process. Nutrient dilution. So many vitamin deficiencies. So many vitamin deficiencies and mineral deficiencies. Again, next one. So the other major implications of this climate change is we don't know how this the abiotic stress factors and biotic stress factors are going to affect the plant community, plant productivity under changing climate. So some studies are showing that elevated CO2 is ameliorating. Some are there showing that there would be no change. Some are showing that I think even the changing climate even aggravates the effects of these abiotic stress factors. Now, so the other phenomena column as I think the long-term growth of this plant under changing climate or elevated CO2 induce a specific phenomena known as the photosynthetic acclimation. So what do you mean by photosynthetic acclimation? I told very clearly here. So the adjustment of photosynthetic machinery towards a particular climatic condition. So this phenomena is always called as the photosynthetic acclimation. Now, so why the plants will always exhibit the photosynthetic acclimation? The major implications of the photosynthetic acclimation is, the first one is I think the altered C by N ratio. What do you mean by altered C by N ratio? Suppose we are giving so much of food to the individual. So the people used to take more food and they will try to build up his body in a random way. There is no proper incorporation. Suppose if you are giving a balanced diet to the person or any individual. So what's happening here? So when we are trying to giving balanced diet to the person, so they will take up all minerals, all nutrients, all proteins and all carbohydrates. So all fats, everything will be in optimum range and which could able to support the growth of individual in a proper way. So here, what is happening in changing climate under increasing atmosphere CO2 concentration? So plants try to use more carbon dioxide, and in this fashion, plant try to invest more nitrogen. So over a period of time, the plant reaches a stage in which the foliar levels of nitrogens are reducing in elevated CO2 grown plants as compared to the ambient CO2 grown plants. So which brings out a simple phenomena known as this altered C by N ratio. What do you mean by so? There is a significantly high carbon content as compared to the nitrogen. So which will induces a simple, due to this one, because of this source to sink imbalance. Source to sink imbalance. What is source here? Leaf. Okay, so the moment carbon dioxide enter into the plant, so the carbon dioxide has to be converting into the sucrose. And sucrose has either sucrose or starch, whatever it may be here. Yes or no? So suppose the plant is unable to accommodate more sucrose or starch molecule. What's happening here? Because due to the limitations in the sink. What are sink here? Sink is nothing but roots, stem, branches. Yes or no? So these all are the sink. Suppose the plants having more sink, plants are having more greater root, and there will be no rooting limitation. What's happening here? The plant tried to continuously, which one? Regarding about this export to the photosynthesis to the respective sink components here. So I think what's happening, so whatever the amount we are eating, whatever the amount they are sequestering, and it will be mobilized toward the respective sink components here. The moment once the source to sink imbalance has been taken place, so which will eventually causes the phenomena called as photosynthetic acclimation. See here. Now, so how do we recognize this? The plant is exhibiting the photosynthetic acclimation. The most important thing is that which one? The increased starch to sucrose ratio. Increased starch to sucrose ratio. So what do you mean by starch to sucrose ratio here? What is exporting sugar? Sucrose. The moment there is limiting in sink, so sucrose again can be reconverting into the starch. The moment starch is happening, when there is a high level of starch, so the high level of starch again inhibit the photosynthesis. Yes or no? Next again, reduced ATP ADP ratio. Again next one, decreased NADPH, NADP. Next one, diminished ribosco carboxylation, RBP regeneration, and AMAX again reduced photosynthetic efficiency. So these all are the different signs by which I think we could able to manifest. We could able to conclude. The plants are attaining which one? Regarding about the photosynthetic acclimation. Now, so these all are the our experimental facility. So we used to simulate the CO2 levels of 2050 year in the University of Hyderabad during my PhD. So call them as open top chambers. Because the top of the chambers were open and try to maintain the almost natural environmental condition except CO2 level. So we try to simulate, we try to pump the 550 CO2 concentration into the chambers, and we try to investigate how the 550 ppm CO2 is influencing the productivity of different types of crops. Now, so here so this is the CO2 analyzer which will control the flow of carbon dioxide into the chambers. So the moment once the chamber has full of 550, it will stop pumping of the CO2 here. So these are the CO2 chambers through which we used to pump the CO2 into the inside the chambers and all these things. So now, so these are the simple basic experimental approaches here. So during my PhD, so I worked on this simple model plant known as mulberry. So why we selected the mulberry as a model plant? Because these are the faster growth, wide genetic diversity, semi-deciduous, and another thing is high responsive to CoPC. Again, next one, it has a lot of medical and ethnobotanical values, and now the mulberry is considered as a which one? Regarding about the potential bioenergy tree species. So these all are the different things we made us to stick to the mulberry plant. Another thing is, most important thing is, the mulberry cultivation is happening in India. Nearly 2 lakhs hectares are cultivating the mulberry in India. So that's why we need to, it's highly imperative to check how this mulberry is going to perform under future climatic condition. Now, these all are the most different types of genotypes what we collected from all over the 5 different countries. From which one? Regarding about the India, Bangladesh, Myanmar, Thailand, and Papua New Guinea. And so this all will be collected and we establish a mini farm in our area in the University of Hyderabad garden. So I think so what could you observe in this from this figure? What could you figure out from this one? See here, I think these are the different types of which one regarding about that I think mulberry genotypes. So this selection 1 Thailand, Mysore local, selection 165, Johari Paki and triploid. So these all are the different types of genotypes. So what did you figure it out? What can you figure out from this picture? So I think you could see here there is a significant difference in stem grid and plant height, leaf structure, leaf quality, leaf color, and all are different parameters, even though they belongs to same species. So people try to observe there is a change in genetic variation. There is a change in genetic variation in performance of photosynthesis. Though even though they all components that belongs to same species, but what happened here? So they could be able to observe certain changes in the even genetic changes here. So here, the moment one the change in photosynthetic efficiency, so which also reflected in the form of yield, biomass yield. So this is how I think people try to figure it out how the plant is able to exhibit a respect to a specific photosynthetic process here. Now, so I think I'm going to show this one, I think very important phenomena. So these are the basic morphological changes what we observed during this mulberry. I think what you figure it out from this, I think you just see here. So do you observe any difference from this? These are the plants which showed morphological changes grown under 550 ppm, and these are the plants which grown under the 400 ppm, 400 ambient CO2. What did you observe? Any difference here? Color of leaf, okay. Apart from that, any? Plant height, number of branches, increased, right? Yes or no? Yeah. The moment when we expose the mulberry to 550 ppm, there was significant rise in photosynthetic efficiency, which could reflected in the form of changes in biomass. Plant height was increased, number of branches, number of leaves, stem grid and root, whatever it may. I think I'm going to show that. So then another important thing is we are going to evaluate how this elevated CO2 is interacting with abiotic stress factors, so-called drought. So what is happening here? So here, these are the plants which were grown under elevated CO2, and these plants could show the morphological changes. So after 30 days of complete water withholding, we have not given any water. To the, I think, this mulberry plants. I think so to check this, how this plant is going to perform even under the drought condition, because the drought episodes are more pronounced under changing climatic condition. So we would like to emphasize, we would like to investigate that how these are going to happen in order to, I think, so we understood that, I think, the elevated CO2. So what did you observe in this? There is any changes in this? Which plants are more healthy here? Ambients are more healthy? See, here more leaf is fallen here. Here all leaves are completely wilted here. What about here? Leaves are still healthy here. Where? Yellowing. It's okay, but here complete dried, right? Here yellowing is there, but here complete dried and they fallen so much of leaf here. Yes or no? So what we observed that, I think the elevated CO2 is try to ameliorate, try to nullify, try to make which one regarding mitigate the impact of drought in, I think, which one regarding about, I think, the mulberry here. Now, so this is the stomatal behavior. So what is happening in case of mulberry plants? So when we expose to 550 ppm. So what did you figure it out from this figure? These are the ambient and these are the elevated. I think in case of ambient plants, more stomata open. The stomatal aperture is also more. As compared to the elevated CO2 grown plants. Elevated CO2 grown plant means the stomatal adaptation has been taken place in the plants which are growing under elevated CO2 conditions here. Then, so these are the photosynthetic physiology what we measured through IRGA, what I showed. what I told here previously. So I think the IRGA can be used to track the changes in photosynthetic efficiency. So this is called which one? I think the photosynthetic rates. Photosynthetic rates. So elevated CO2 grown plant showed higher photosynthetic rate without drought as compared to ambient CO2 grown plant, but the drought imposition reduced photosynthetic efficiency, but the efficiency was reduced more in case of ambient CO2 grown plant as compared to the elevated CO2 grown plant. Again, see here, reduced metal conductance. I think I told very clearly. So the plants which are grown elevated CO2 try to maintain higher intercellular CO2 concentration. I think lesser transpiration in elevated CO2 grown plants, then followed by the, I think, more water use efficiency. And so these all are the different parameters which could always take in place to assess the plant performance under changing climate. Assess the plant under the changing climate here. So now, so these are the, I think, FU by FM. What is FU by FM? Maximum quantum yield. Okay, so this is the changes in photosystem to efficiency, then followed by the yield, non-photochemical quenching, and relative water content. What is relative water content here? So in order to assess the relative water content, we have to check this leaf water status. See here, there is no much change in the control condition, but when we are giving the drought, what's happening here? So I think the elevated CO2 grown plants are trying to retain more water because there is a less transpiration rates. As compared to the ambient CO2 grown plant, I think, so these are the different curves. This is the A by CI curve, A by Q curves, I think increasing photon flux density and increasing, I think, with respect to CO2 assimilation rate. So plants which were growing under elevated CO2 tend to having higher photosynthetic rates even under both well-watered as well as drought condition as compared to the ambient CO2 grown plant, which will further reflected in the form of which one? Regarding about this, the apparent quantum efficiency, light compensation point, and A saturated, which clearly indicating that increasing atmospheric CO2 levels are try to minimize the photorespiration, and which eventually causing this more photosynthetic efficiency. I think this is the very important fast chlorophyll, OJIP chlorophyll fluorescent transient. I think so it will always track the flow of electron from photosystem 2 to, I think, photosystem 1, including from the water splitting complex, efficiency of the water splitting complexes. So which can be divided different regions. O region, I think this is the this part will emphasis on OJ region, and this part will emphasize on JI. This part will emphasize on, I think, IP region. So which will be always tells about the flow of electron, how the electron flow has been taken place into the in case of different consequences here. Now, so these are the results what we observe. See here, I think these are the raw chlorophyll fluorescent transients, and these are the which one regarding about this, I think the normalized things. I think I don't want going deep in this because I think you may get again confused here. So I will just summarize these consequences here. And this normalization we did for this the observing of this a specific type of band known as, I think, L band. So what is the major concern about L band? Why do you look for this L band? So see, these are the well-watered plants. We don't observe any L band. But the moment once you try to impose the drought, so then which will always able to produce a unique peculiar band known as L band. This L band will majorly emphasize the connections between light harvesting complex as well as the photosystem 2. Suppose there is a rise in amplitude of L band. There is a high, suppose see here, which one here? This is the pink one. So pink means what's happening? More L band here. So these are ambient CO2 grown plants with a drought. So if there is this is a inversely proportional. More L band, less connection. Less L band, more connection, more stability. Yes or no? So here, so what's happening here? So there is a rise in amplitude of L band in ambient CO2 grown plant, which clearly indicates that I think this plant doesn't have proper stability. There is a destabilization of which one regarding about that, I think the light harvesting complex as well as this the photosystem 2 complex. Then we also tracked for the another type of band known as which one regarding about the K band. So the K band which majorly emphasizes this the efficiency of water splitting complex. See here, the moment once we observe this rise in amplitude of water split K band, so which clearly indicates that inefficiency of oxygen evolving complex. oxygen evolving complex. So here, so what's happening? So that's why I think so this ambient CO2 grown plants doesn't have the proper oxygen evolving inefficiency of this I think the oxygen evolving complex. So these are the very important parameters we could able to assess the efficiency and stacking and stabilization of this I think chloroplast membrane, and so which eventually causes the simple which one, the formalities regarding about in case of the plant performance here. Then the changes in chlorophyll fluorescent transients were further translated into Gyp test parameters. So there are so many Gyp test parameters here. I don't want to go in deep into that. I think I specifically focus on this which one regarding about some important parameters which tells about the photochemical quenching as well as non-photochemical quenching. So what do you mean by photochemical quenching? And what do you mean by non-photochemical quenching? Suppose one plant is there here, two plants are there. One plant could able to take up 80% of light and 20% light is emitted in the form of heat. Then another plant could take up this 60% of light and could emit in the form of light 40% in the form of light, heat energy. So which one is more efficient? Plant A. So because this plant could able to exhibit more photochemical quenching, so that's why the moment one it has started performing more photochemical quenching, and the flow of electrons will also more. Okay, so here now, so these are the more important parameters. I think Fe by Fm, Fe by F0, electron transport for cross section and performance index. So these are the parameters which tells about that how this the plant is going to show, this plant is going to exhibit the process called as I think the photochemical quenching. So what we observed that at both elevated CO2 grown plants at well-watered as well as drought condition, they showed the rise in the parameters like Fe by Fm, Fe by F0, electron transport chain and performance index, and with concomitant reduction in the other parameters which related to non-photochemical quenching, which one regarding about I think F0 by Fm, Kn, dissipation per reaction center, dissipation per cross section, so which clearly indicating that the plants which were grown under future atmospheric CO2 concentrations are having more photochemical quenching in case of mulberry, but it is not applicable to all other plant species. I told very clearly the plant responses are highly specific, and even the plants they belong to same species are going to show some difference in photosynthetic efficiency, and we can't apply the same principles, we can't apply the same response toward this another type of plant. Suppose we can't compare the rice plant performance with mulberry plant performance. You can't compare the mulberry plant performance with another X plant. So here, so these are the plant responses are highly unique, and that's why we are trying to screen very important and very potential potential crops. So here, so which clearly indicating that the elevated CO2 improve the performance of these plants, improve the performance of photosynthesis in case of mulberry plant, so which will eventually reflected in the form of this increasing biomass yield. You already seen. Yes or no? Now, I think this is another story, very important story here. I have been telling that there is a less membrane destabilization, means there are more membranes are more stable, more efficient, and all these things is happening in case of this elevated CO2 grown plant. Now, I am trying to correlate with this I think antioxidant metabolism. So what is antioxidant metabolism? Do you have any idea about antioxidant metabolism here? I think it is ubiquitous. Just look at animal cell, look at plant cell, look at algal, anywhere. Antioxidant metabolism is universal, ubiquitous. Without this, I think there will be no existence of any type of component. Yes or no? The moment we start feeling stress towards something, our system try to combat that stress through a signaling process called as ROA signaling, reactive oxygen species. Suppose you feel more stress. Whenever you feel more stress, our cells try to produce more ROAs. What are the example of ROAs? Oxygen radicals, hydroxy radicals, H2O2, singlet oxygen, doublet oxygen, whatever it may be here. So, which will eventually degrade the cell structure and function. How these ROS are going to degrade here? So here, the ROS will always able to induce oxidative stress, so which will oxidize lipids, amino acids, proteins, nucleic acid pigments, and which will eventually cause this cellular damage, and finally the cell will subject it to death. That's why the cell has defense mechanism. The cell has developed some defense mechanism to combat, to sequester these reactive oxygen species. Suppose the production of ROS is equals to the sequestration of ROS, and there will be no problem. Plants will happily survive. The balance has moved towards which one? That is more ROS. The balance has been shifted towards more ROS, then which induces oxidative stress and which will eventually causes this the plant cellular death. Are you clear? So more ROS, more oxidative stress, more stress effects. Less ROS because efficient antioxidant metabolism and less oxidative stress, and finally plant will survive happily. You don't have any problem with that. So we want to track that, how this antioxidant metabolism is going to be changed in case of plant cell, in case of the plant cell. Now, so we have seen and we perform the, I think, different types of activity under elevated CO2 with respect to drought as well as well-watered plants. Surprisingly, we observed that, see here, the dotted lines are ambient CO2 grown plants, and the thick lines were elevated CO2 grown plants, and we did not observe any elevation. We did not observe any elevation in the antioxidant metabolism until the 30 days. What this indicates that? Why plants are not coping up? Whenever you feel hungry, then you will eat, right? You don't feel hungry, you don't eat, na? Yes or no? Because the plants are not feeling stress. Then why do they able to produce more amount of antioxidants? They are more healthy. Are you clear? So the moment the plants are more healthy, then they are saving their energy. They are storing their energy. They don't want exhibit, they don't want perform the unnecessary activity. So here, but the ambient CO2 grown plants are feeling more stress, and which eventually causes, I think, so what's happened? There is a surge in antioxidant metabolism. And until the 15 days of that, but the ambient CO2 grown plants could not able to handle this ROS, and which eventually causes this death of plant cell through the oxidative stress. So here, this is how I think what's happening here. But here, these are the ambient CO2 grown plants, even they could able to handle the stress even after 30 days of the stress. After stress. So now what's happening? So this is happening with all different types of enzymes here. So here, there are different enzymes are there: superoxide dismutase, ascorbate peroxidase, catalase, glutathione reductase, GPX, MDHAR. So these all are the different enzymes. They could take part in the process of antioxidant metabolism, so that which could able to sequester the ROS and finally could able to neutralize this ROS component. Then we have seen similar result in case of transcriptomic analysis. So we perform the QRT-PCR analysis, that is the gene expression studies. So the gene expression studies also, we have also observed the similar changes. There was a significant rise in transcript levels of different antioxidant enzymes in case of ambient CO2 grown plant, even under well-watered conditions. Then the similar trend was observed even after 15 days, but after 30 days, the ambient CO2 grown plants could not able to handle the stress, and finally, so they lowered this the activity of all type of enzyme because the cells were died. So this is happening with regarding about, I think, the changes in the gene expression of different types of enzymes. Then I think these all are the non-enzymatic antioxidants: ascorbic acid, total phenolic content. I think the proline. So do you have any idea about proline role? Have you heard about proline? What is proline? Proline is amino acid. Yes or no? So what is the role of proline in abiotic stress? The proline, the quantity of proline will always tells us the stress level, stress intensity. More proline, more stress. Less proline, less stress. Yes or no? See here, now what's happening here? So here, ambient CO2 grown plant showed more proline quantity here. Indicates that they are feeling more stress, which could be reflected in the form of H2O2 levels. So they are accumulating more H2O2 because they could not able to sequester ROS. Inefficient antioxidant metabolism. Though finally, so I think another important for MDA. What do you mean by MDA? So what is the indication of MDA? Melandialdehyde. Melandialdehyde. So this is the parameter which will always tell us about lipid peroxidation. So I think you know very well all members are made up of lipids. So the moment there is a rise in MDA quantity, which clearly indicating that the lipids are more subject to oxidation. So this is why the plants are grown under elevated CO2 showed more healthier as compared to ambient CO2 grown plant, and the destabilization has been taken place more in ambient CO2 grown plants as compared to the elevated CO2 grown plants. Are you clear? So these all are the different consequences what we observed in case of this consequences. So here I think these all are the different biomass. I think so these are the morphological differences. I think DAT stands for days after treatment. Days after treatment, then followed by the here, this is 90 days, and these are the morphological changes after 360 days after 1 year. And so this is the different types of biomass what we observed after 1 year when we harvested the plant. So which were grown under which one? Under elevated CO2 grown plants, and as well as the ambient CO2 grown plant. I think you must have asked about that. What is SRC? Do you mean what is SRC here? I think your short rotation CoPC. What do you mean by CoPC? Suppose outside one tree is there, neem tree is there. If you cut down the plant, to grow up, it will take years. Okay, but when you cut down the plants for mulberry, it will resprout within a span of 3 months. So we used to harvest the plant for every 3 months, and we used to generate the biomass for every 3 months. And because our motto is able to produce the more biomass for production of more energy. More bioenergy. So this is how what is happened that the SRC1, SRC2, SRC3 and SRC4 yearly we used to harvest the mulberry plants for 4 times. 4 times, and we used to generate a lot of biomass, and this biomass can be able to reflect. See here, this is the biomass what we generated in elevated CO2 condition after 1 year, and this is ambient CO2 grown plant. We just see here the number of main primary branches, and here these are the major differences regarding about that. I think the plants grown under elevated as well as ambient CO2 grown plant, and this is the root biomass what we observe. And we calculated carbon sequestration. I think the ambient CO2, elevated CO2 grown plants almost, I think, see here, this is the plant biomass after 1 year, nearly 20 kg in elevated, and this is the, I think, 10 kg. How much difference? 50%. Okay, so here we calculated CO2 concentration, and we also calculated the amount of CO2 sequestered by this dry biomass plant. Then, so finally, we could able to extrapolate towards which one regarding about that. I think toward this per hectare. I think so. These are the plants. I think similar things is happening regarding about that. I think so. These are the plants even after 3 years. So we continued our experiment for 3 years. That was first 1 year, and we prolonged this experiment for 3 years. So we would like to know how much how the plant is going to tolerate for this. How long it can tolerate. How long it can perform this the photosynthesis. Still, we could able to see. I think you can see clearly visible this difference here. I think this ambient CO2 grown plants and elevated CO2 grown plant, still it is performing better per photosynthetic efficiency after 3 years. So after 3 years means how many harvests we have done? We did how many harvests? 12 times. We harvested the 12 times. So again, we calculated this biomass. I think after 3 years, the biomass could be around nearly 85 kgs per individual plant, as compared to the, I think 35 around nearly in the elevated CO2 grown plant, and the amount of CO2 sequestered by this when we extrapolated towards per hectare, nearly around the 400 megagrams per, I think, tons. 400 tons of CO2 has been sequestered by this mulberry per hectare. So what we are indicating that I think these plants are more efficient. So, but what we observed surprisingly that, so when we track the photosynthetic efficiency, changes in photosynthetic efficiency through A by Ci curve, so I think what I told here, the rise in amplitude of these graphs. So there were, after 6 months, the plants showed better photosynthetic rates. I think we don't have any observance. We didn't observe any acclimation, but the trend was changing when we are moving toward this. When the years were progressing. See here, I think after 1 year, I think the elevated CO2 grown plants could not able to exhibit a proper, I think, a proportionate photosynthesis as compared to the ambient CO2 grown plants. Again, so this is happening after 3 years, and so that's why. So when years were increasing, so which eventually causes this the down regulation of photosynthesis. I think we had a lot of experiment, did a lot of experiment not to confirm all these things. So I think the ultimate one is, I think there is a significant dilution of nitrogen. Significant dilution of nitrogen. So I think so this is Vc max, J max, and A max. So it could be further reflected as we seen here. So I think so there was a higher in 6 months in elevated CO2 grown plant, followed by the reduction in after 1 year and 3 years. Again, so to confirm this photosynthetic acclimation, we perform this the comparative proteomic analysis, and in this we have seen the different types of proteins which were showed some differential expression, and which are related to cell signaling, cell wall biogenesis, energy metabolism, fatty acid biosynthesis, nitrogen metabolism, photosynthesis and protein metabolism, and different protein. So I think nearly we showed 70 different proteins. 70 proteins are showing the differential expression. Now, so but we are more focused about photosynthetic protein. We wanted to correlate these changes in A by CI curves with the protein expression. So what we did, so we have done this comparative proteomics, and we identified different types of proteins which are related to photosynthesis are showing this down regulation here. So this is the Rubisco large subunit showed down regulation. There is a significant lower in elevated CO2 condition as compared to ambient CO2 grown plant. Again, next one, the glutamine synthetase, the most important enzyme which is take part in nitrogen metabolism, is higher in case of elevated CO2 grown plants. Rubisco active is also very high, and finally, some other proteins like oxygen evolving enhancer protein, pseudoepiose 1,7 bisphosphatase, and phosphoribullokinase. So these all are the different proteins, and they showed this down regulation. And so which is having some important consequences. So which clearly tells us that so the mulberry plant, which are grown at elevated CO2 after 3 years, starts showing this down regulation. Starts showing the down regulation process here. Now, so with this, I think I could conclude. So I think. The climate change is more prominent. We should be very cautious and very conscious about this crop productivity, and which are going to have a lot of negative impact on our society as well as human health, and that's why we should be very careful regarding about this thing compared to all these things here. So I think I would like to congratulate this with this all organizing committee, and I would like to say about my teacher, Professor Rajgopal sir. I think he's our teacher. So though we are not direct student of Rajgopal sir, but I think we admire him, we love him, and we respect him a lot. And he's a very good human being. He's helpful to everybody. Suppose if you want to work under him, you just write a mail. He will accept you, come and work with me. You don't have any problem with that. Such a nice person, such a wonderful human being. Okay, so this would I think I will conclude my session. Yeah, any queries, please let me know. I'm happy to answer your queries.
Sir,
yeah,
I have one question.
Yeah.
So, is there any effect of ambient and elevated CO2 in silkworm production of the silk?
Yeah, because that would be another PhD work. Huh? So we didn't check it. We didn't use that leaves for how this. Actually, we would have any collaboration with somebody else would have tried that, but I think unfortunately we didn't do that. Actually, definitely there would be some effect over that.
Maybe you are thinking positive or negative thinkings are there.
So because this plant is performing better, so I am hoping that positive response. So but depends upon the plant age also, depending upon nutritional status. If you are starving the plants, definitely we will be having some negative impact. If you don't starve the plants and we are feeding them happily, then plant could able to perform better, and it might be having some positive impact over that.
Sir, mulberry plants for the biomass growing. So water is requirement is needed more na actually.
Yeah, that's what. So not needed much actually. I think we used to give water for every 10-15 days like that in our field. But it's a water demanding plant, no doubt about it. Because these are fast growing tree species, right? Mm-hmm. So we have to always give some. But in some area, this plant is growing in a right-fed manner through rains also, rain-fed manner also.
Thank you, sir.
Any?
So I have a question about the quantification of
ROS?
Colorimetric?
Calorimetric, yeah.
Colorimetric.
Yeah, calorimetric.
What about enzymatic activity?
We did calorimetry only.
For everything?
Yeah, everything calorimetry only. Not spectrophotometry, not calorimetry.
Full spectrum or
Yeah, full spectrum. So many protocols are there in literature. We can go through it, no problem. This RT-PCR we did through, I think, real time, and the remaining all are calorimetry, spectrophotometry. Any queries? I'm happy to answer your queries.
Anybody is having
Yeah,
any question?
come.
Sir, the increase in carbon dioxide levels, are they good for the plant?
Yeah, see, increasing carbon dioxide level is good to some extent. Okay, but the way we are pacing now, I think it is going beyond our limitations. For I think for some time increasing atmospheric CO2 to some extent it is beneficial. But you know, I think I told here the moment once a rise in CO2 level, there is a rise in temperature also. There is a rise in prevalence of more droughts. I think there is lots of variation, lots of changes in climate. So which eventually all combine together, and instead of having positive impact, these plants could able to I think climate having showing more negative impact. When we are integrating individually, it's having positive impact, but when we are integrating all other factors, it's more effective. Clear?
Because you also said that over the 3 years, the nitrogen content
Yeah.
is diluted.
Yeah.
So that may also have effect on the plant.
Yes. Because of that, because the nitrogen dilution may not. That's what he was asking question that. So how these plants are going to fetch? Suppose if you use these leaves to raise the silkworm, so how about their quality of silkworm? Because definitely the moment one we face, the moment one you see that, because we have already seen that, there is a lot of nitrogen dilution. There is only increasing biomass. There is only increasing quantity, but there is no quality. We need quality, right? So this how I think it will be cumulatively affecting this total overall growth and performance. Are you clear?
There are no questions. We need to thank Dr. Madan for wonderful talk. It is very nice.
Thank you, Venkat. Thank you so much. And
You can interact with sir also. He will be here for stock. Professor Prakash sir will give the next talk.
Okay, so the very last lecture. So the previous lecture, I forget to add some important data and I explained without the figure. Maybe as I hope you remember that PET YCF7 subunit encodes a small hydrophobic subunit of cytochrome B6F complex with a single transmembrane helix. Okay. So the absence of this gene and the absence of this PET-L subunit, B6F complex is still functional, but at a low activity and low stability of the complex. So this is the data I talked, but I show you the wild type cells and YCF7 clone 3 cells. This is PET-L deficient mutant cell, and the one is the cell of exponential phase, so actively where the cell actively synthesize proteins, and second is early stationary phase, so almost translation activity is reduced, and third, 3 is the stationary phase. The cell do not synthesize most proteins. Okay, and then we measured fluorescence induction kinetics. Show you that this decline, the fluorescence peak, indicate the activity of cytochrome B6F. The wild type, the activity stay the same, right? Similarly, the mutant, the stationary phase, it shows a little bit the activity of decline. Second, also still, but in the stationary phase, the activity of B6F almost absent. And if we analyze the protein level by immunoblotting, wild type, the level of the accumulation of cytochrome F, this is a measure of the level of cytochrome B6F complex in the chloroplast is almost the same. A little bit decreased in the stationary phase, but it's almost the same. But in the mutant, it goes down significantly reduced. You don't see, scarcely you see the signal. So stationary phase, accumulation of protein of B6F complex was almost undetectable. So this means that the stability of the B6F complex was significantly affected. But in the young cells, so protein synthesis is active, so certain level of B6F complex accumulate and functional. So PET-L is not essential for activity of cytochrome B6F complex, but important for the stability of the B6F complex. So this is a crystal structure of cytochrome B6F complex. This is a dimer structure, actually. It's not monomer. Because the iron sulfur FES protein is this yellow. This was dropped off after purification from the B6F complex without PET-L. So this is yellow one. And interesting thing is I should say, this because B6F is dimer because this monomer is all the subunit is present as a monomer in the monomer, but this iron sulfur FES protein associated with this monomer comes from the other monomer, and this one comes from this. This is called domain swapping. So that's why B6F complex must form dimer to express function, activity. So this green, there are three small subunits. PET is one of them, and single transmembrane helix, like this. So without this, probably the peripheral region of this complex is directly exposed to, so without this, it's exposed, so probably proteinase more easily attack B6F complex. So that makes the complex unstable in the absence of PET. So this is our conclusion, okay? So the next topic is the site-directed mutagenesis of PSBA and PSBD genes. These two genes are encoding photosystem 2 reaction center subunits. So we introduced how to make a point mutation. You change one or two nucleotide to change the amino acid, replace the amino acid, and to see what happened to the protein. So this is a photosystem 2 complex. It's complicated, the crystal structure, and this also forms dimer. So photosynthesis complex like dimer structure somehow. And this is a reaction center component. You see, so this only mostly show the cofactors involved in photochemical reaction, okay, and electron transfer reaction. So if you look at this, the monomeric PS2 complex shows a symmetrical structure. Maybe I told before the symmetrical structure of photosystem 1 and photosystem 2, and these two complex has similarity in structure. So the D1 protein, that is a PSBA encoded D1 subunit, and PSBD gene encode D2 protein. And these two proteins are sequence is similar, has similarity. So we call the reaction center as heterodimer. And in this heterodimer, component of redox component are located. PD1, this is a chlorophyll pigment, chlorophyll associated to D1, and pigment chlorophyll A associated with D2, form a chlorophyll dimer, something like this. And this is chlorophyll D1, chlorophyll D2. This is named accessory chlorophylls. And this is phyophytin, symmetrically located. Phyophytin D1, phyophytin D2. Okay. Phyophytin is the pigment derived from chlorophyll A, but the magnesium in the center of chlorophyll A is lost. So that without magnesium, This is called phycin. Okay? Very similar to chlorophyll, but no magnesium in the middle here. And next is QA, bound plasquinone, QA, and QB. Also, these two bound plasquinone is located symmetrically, okay, in D2 and D1. It's a little twisted. Okay, and then from QB to plasquinone pool, the electrons are transferred. So, and also the donor side, there is a tyrosine residue on D1 protein, PSBA subunit, is electron donation, electron donor. And then manganese cluster, mangan 4, calcium 1, oxygen 5, form a cluster like this, like this. So this is the site of catalytic site of water oxidation. So electron extracted from water transferred to tyrosine, Z, tyrosine residue on D1 protein, and P680, this is a dimer. And then chlorophyll D1, phycin, QA, and then QB. And the QB is when reduced by 2 electrons and accepted 2 proton becomes plastoquinol. Then released from this site, it go into plasquinone pool, and oxidized plasquinone is replaced and bind to here. They accept another electrons. This is a reaction, series of reaction in photosystem 2. So now I'm going to focus on the oxygen evolving site. So this is the manganese cluster. Okay, is associated by several amino acid residue on D1 and CP43, PSB C or PSB A and PSB. So this amino acid is the bind of this manganese cluster. Okay, and then oxygen evolution. How oxygen evolution occurs? Did you learn already in this seminar? Not yet. Okay. So water, 2 water molecule is oxidized 4 times to produce 1 oxygen and 4 proton. Okay. 2 H2O oxidized 4 proton and 1 oxygen molecule. So 4 times oxidation means 4 times extraction of electron. Now, oxidation means the losing electron. So this manganese cluster has 5 different states. That is named S0, S1, S2, S3, S4. And if you start, actually it starts from S1, but if you think to S0, the 1 photon comes and then one reaction occurs, then 1 electron is extracted and 1 proton is released. And the second photon comes, so it moves to S1 to S2, 1 electron. And third photons transfer S2 state to S3 state, and then 1 proton and 1 electron. And last, the fourth photon comes, and then 1 electron and 1 proton is extracted, released, and then finally reach to S4, and then without light photon, it changes from move to S4 to S0. So then a proton, 1 proton is released, and then 1 oxygen molecule is released. This is a so-called S cycle, no S cycle. So this is the number of flash, excited cell, and then this is the yield of the oxygen. So if you, the dark adapted cell is illuminated by a single flash, no oxygen is evolved. Okay. The second flash, a little bit. And third flash, it reaches the peak. And the fourth, it goes down again. Fifth, sixth, seventh, again, go up. And then this oscillation is gradually dumping, smaller, smaller. Okay. So if you look at the peak of oxygen evolution, this is the first one is third flashes after when the third flashes were excited. But next following a peak is interval is 4 flashes. Okay, that's why this model was proposed. 1, 2, 3, 4 flashes, and then go back to the S0 state. So the cycle is 4 because 2 water molecules need to be 4 times oxidized. Okay? But the question is why the first peak of oxygen yield, high yield, is reached in not after the 4th flash, but in the 3rd flash. Okay? This is a question. But this was a very long time ago in the 60s. This experiment was done, and this model was proposed in the 60s. So at that time, the people were thought that the 4th cycle is as expected because 2 waters must be oxidized 4 times to produce 1 single oxygen. But why the 3rd time? Now, why it's damping this oscillation? So if you think so seriously, it's difficult. But if you think easily, okay, starting point is S1. Okay. Did you think so? It's easy. No, if we found a model, if we propose the model, oh, that's easy to understand. Okay. So starting point is here, and then 1, 2, 3 flashes, and oxygen comes out, and then second round, 1, 2, 3, 4. You need 4 flashes. That's it. So this is a cycle. But this damping mean the flash is sometimes not always, always it does not hit 100% of photosystem 2, not completely. And also the flashes has some width, so very small number of photosystem 2 is hit by 2 times by single flash. Okay, the beginning of single flash, its reaction occur, and the end of single flash, this may happen. Second reaction may happen. So that is called double hit. And some PS2 reaction center couldn't get any photons, so that is called miss. So this double hit and miss occur about 10%. So if double hit occur, if it starts from here, one single hit and double hit, then oxygen comes out. So we can explain this result easily. Our question is, when the proton is released at the manganese cluster, oxygen evolving site, it is a little bit inside the protein. So how this proton is released, transported into luminar site? Then we did a site-detected mutagenase that may be involved in this proton transport pathway. That is encoded by PSBA, that's coding D1, and PSBD, that's coding D2 protein, 2 reaction center protein. And as you can see, PSBA is encoded in the inverted repeat region of chloroplast DNA. So PSBA gene, there are 2 genes in single chloroplast. So we have to introduce a mutation. If we want to introduce a mutation in PSBA gene, we have to change, similarly, both genes, right, in the inverted state. But it's possible. It's easy. It's the same efficiency. Changing a single copy region, inverted repeat region, we can easily change because the chloroplast has a copy correction activity. So this inverted region is maintained precisely the same, maintain the same sequence. So if you change one of the gene, automatically, if you have a selection pressure, both gene, you can introduce the same mutation into the both genes. So this is from the crystal structure. This is luminosite, this region. And then manganese cluster is here. So we expect that we want to know how the proton released by this water oxidation reaction, how this go outside. This means proton channel. We think that basically, according to other proton channel model, we think acidic amino acid may be involved in proton channel. The proton is so small, so it may show a carbonate or single proton itself cannot exist because proton is a hydrogen nuclear, right? So, but proton can transport it by exchanging the proton between proton, for example, acetic acid, the side chain of acidic, for example, carbonate, carboxyl group, or water. Water can also transfer proton. The proton, because water can accept the proton, bind the proton. So, but we expect this residue. This arginine is alkaline, but alkaline side chain also can exchange a proton. So these are all acidic amino acids. So if these are involved in the proton transfer reaction or proton channel, if we change this amino acid, the PS2 photosystem 2 activity or oxygen evolving activity may be affected. So to see if this happens or not, we changed this amino acid. So the question is to which amino acid we should change. We don't know. So the Dr. Hiroshi Kuroda, he was working with this. He changed, okay, to 19 different amino acids. Usually, we don't do that, but he did. Okay, so he changed this amino acid, possible change. So B1, PSBA, aspartic acid 61, glutamate 65 here, and glutamate 312, arginine 334, glutamate 302 here, and glutamate 323 here. Okay. 1, 2, 3, 4, 5, 6. And D1 glutamate. Okay, well, this is D1, PSBD. So he made 1, 2, 3, 4, 5, 6 site to change. Okay. To 19 different amino acids. Okay. So for this project, 1, 2, 3, 4, 5, 6. So 6 means 6 times 19. So 114? 114.
160.
19 times 6.
6 is 114.
114, right? 114. 114 mutants he made.
Sir, which technique he used for mutation? Which technique he used? Which technique he used for mutation?
Chloroplast transformation, and to make the transforming vector, sometimes you can make the primer with regenerated, but then you have to screen many to find that maybe 18 we can find, but final 19 mutant, it took many times that may happen. So he made 19 different oligonucleotides to make, so one by one. He didn't use a sophisticated way, but it's a very classical way, but very, how to say, certainly he gets 19. Okay, this is the result. Spotted on agar plates, the cell culture, under the condition of photoautotrophic condition. So if this mutant grow photosynthetically or not in the beginning, okay? So this aspartic acid 61 very close to manganese cluster. The growth was significantly affected compared to others. No? This is control. Control means aspartic acid. Okay? And glutamic acid. Cell could grow because this is, as expected, because this is acidic, the amino acid. So still, the side chain of the carboxyl group can exchange the proton probably. This is interpretation. So that's still a proton channel works. So that is our conclusion. But so others didn't grow. So this carboxyl group is very essential for proton for the proton channel. But as you notice, why cysteine? No, cysteine worked. Cysteine is not as acidic acid. It's not regarded. If you look at the textbook, it's not. But cysteine has SH side chain, right? So H. This can be deprotonated. Usually we don't think so. No, cysteine can be involved in the exchange of proton. But okay, of course, glycine, alanine, valine, leucine, isoleucine, threonine, asparagine, leucine, this has no side chain that can exchange proton. So, okay, so then cysteine. This result we could get because we changed this residue to 19 different residue. Because usually for the engineering of, if you want to engineer the the protein, usually we engineer. In this case, aspartic acid to asparagine or glutamic acid or glutamine. Okay? Probably that's it. Or other negative control, alanine, something like that. It's small side chain, hydrophobic. Okay? So, it was a lot of work for him to change, to make 19 mutant, but because of this hard work, we could find interesting results, finally, okay? And then others, this residue, a little bit far, located a little bit more distance. And then 3 to 1, this, and this. So if you look at here, it's this also a stronger effect, okay? This one, you don't see much growth here. Aspartic acid, it's acidic acid, acidic amino acid. So this also substitute for partial function. And histidine and lysine, a little bit. Histidine also has a protonation and deprotonation activity. And lysine also, it's an alkaline side chain, but it seems to work. But here, even glutamic acid 312 is a little bit more separated. So even hydrophobic, the residue, alanine, glycine, valine, leucine, soleucine, no, you see slightly growth. And here, this almost all the residues can be replaced. And also 3 to 3 here also. So for the proto channel, this region was very essential, but in the downstream region is less essential. Can be it's also possible bound water molecule can involve in proton channel. So this part is essential, but this part is less essential. So it's not so strict channel. There are kind of there are many alternative way. If the proton is moved from here, then proton can move more freely, more relatively freely. Okay. So this we could find. So this is based on the high resolution of PS2 structure, so that this experiment becomes possible because of the structure. But only the structure, it's still some open question remains how the proton channel. You can predict the channel, but you need evidence, experimental evidence. So this site-to-death mutagenase to change specifically amino acid is powerful approach, powerful tool, I should say. And also, if you look at this mutant that didn't grow, of course, which suggests more than one possibility that, of course, the channel didn't work. But if the protein becomes unstable and didn't accumulate, of course, mutant do not grow photosynthetically. So this possibility still remains, okay? So it's too early to conclude. So we tested.
Sir.
D1 accumulation, PS2 accumulation, black bar, and oxygen evolution. This is a activity. Of course, if the proton channel didn't work, oxygen evolution should be inactive. So this is also making many mutants was a lot of work, but analyzing these many mutants are also very hard work, because for this you have to grow including a control strain, a 20 strain you have to grow under the perfect condition. Maybe someone who experienced growing a chlamydomonas, and maybe you can understand how difficult to handle with the 20 strain under the perfect condition. The cell must be, we have to collect the cell from the exponential phase, logarithmic phase, not saturation phase. Okay, the mutant becomes unstable in the stationary, could be unstable in the stationary phase, as I showed before on YCF7 PETL mutant. Okay, if you change the mutation, amino acid, it is general that the protein becomes less stable. That is natural. So we did this experiment. So it's certain amount of PS2 accumulate here, except for tyrosine and tryptophan mutant. So tyrosine and tryptophan mutant didn't grow because PS2 didn't accumulate. But we don't care, because proline and tryptophan may not be involved in the proton channel, because there is no proton association and release. And the B is glutamine 69, 65 is here. Only proline mutant was reduced to 20%, but others are very, very much accumulated. So proline didn't grow so well. We can say because this is accumulation was low, but the proline is hard to think that it's involved in proton channel. And this 331 glutamate is also proline affect accumulation, but others accumulate very well. And this also shows some oxygen evolution. Okay, so that's why they grow better. Proline didn't accumulate, and also no oxygen evolution. The proline, no, nothing. 312 is this one. But others grow, no. And also some oxygen evolving activity. Of course, aspartic acid is higher than other mutants. And somehow, strangely, tyrosine also evolved more oxygen. So tyrosine grow well. So this is consistent data. So this is the way to analyze the phenotype of the mutant. So we also measured more specific partial reaction of PS2. Oxygen evolution is whole PS2 reaction. So maybe here you didn't experience in the laboratory course of the thermoluminescence. The PS2 activity can be measured by thermoluminescence. So I just want to show the technique here. In the cell, if you incubate in the dark, the oxygen evolution S-state evolving complex manganese cluster is stay S1 mostly, okay? Not S0, S1. And then QA is oxidized state, okay, in the dark. Then cell, you illuminate the cells with single flash. So one reaction occurs, so S state move to S2. And then electron goes through like this and then go to QA. And then here, if you add the inhibitor that block the electron transfer from QA to QB, the electron stops here, and then after flash excitation, you immediately free the cell. Then this S2 state and QA minus state is stabilized in the frozen cells. Okay. Otherwise, the high energy state QA minus donate electron to manganese cluster to make the PS2 the original state before excitation. Then you slowly increase the temperature. Frozen cell to slowly increase the temperature. Then this back reaction to QA to S2 state occur. Okay. To this is a back reaction. So for this back reaction, you need a barrier of the energy, okay? Activation energy. Activation, so it's not instantly go there. In a low temperature, this state has no energy to overcome this activation energy. But if you increase the temperature, from the temperature, it's get the energy for the activation. So slowly, if you increase the temperature, then at certain temperature, this reaction occur. And then if you measure the temperature that occur this back reaction, you can measure know the condition of S state and QA state. Probably in this mutant I present here is this state must be affected by amino acid substitution, not this state. Okay. This is called Q band, and there's another B band without an inhibitor. So this is another. I'm just today talking about Q band. This is a thermoluminescence Q band. Okay. So control. If you excite the cells with flash, if you don't excite cell flash, no signal as a negative control. And you excite the cells with a single flash, and then freeze the cell, and then increase the temperature from 0, this shows 0, 10, 30, 50 degrees centigrade. This means that this supply energy for activation. Okay? So control strain shows that this peak about 10 degrees. And glutamate also very similar. Glutamine, a little bit shifted, but very similar. Cysteine, serine. Serine has a little bit broad band. So some parts are very similar, but the other part of PS2 need more activation energy. This data shows. FUD7 is PS2 deficient mutant to show that these signals are derived from PS2 activity. Okay, this is negative control log. Without PS2, this signal can't be detected. So this kind of experiment we can do without breaking the cell, the intact cell, using the intact cell.
What is that FUD7? FUD7.
FUD7 is PSBA gene deficient deleted mutant. The PS2 minus mutant. So this is very often used for transforming a PSBA gene because the chloroplast of this mutant has a large deletion of PSBA gene. So if you use a host, this strain as a host, it's already major part of the PSBA gene, not entire part, but most part of PSBA gene are deleted. So if you transform, that means a complement. So you don't need to worry about the remaining wild-type genes. So this is very convenient. So this type of experiment we can do. So this is I show only few data because if you show 90 different data, it's just confusing you. So look at this again. Cysteine, glutamate grow, but for example, serine. Serine is, do you think cysteine and serine are similar amino acid? Side chain, OH is serine, SH is cysteine. So it's a little similar, right? But we had a very big cysteine and serine, no, very different. And if you look at the cysteine and serine, both are very much reduced, but the cysteine has a few times, 4 or 5 times more oxygen absorbing activity. Okay, so why this happens? So that is our question. So this thermoluminescence data show that the signal of serine mutant, the mutant changed to serine, has a rather distorted signal. So if the QA is not affected by this mutation, S state, manganese cluster, structure and function are more affected compared to cysteine mutant. Okay, so this we just wanted to try to explain, interpret the phenotype. So this part is a little bit I forget to, I didn't include some important data. Oh, sorry. No. I also failed to include the data. So this is to explain this phenotype. We collaborated with bioinformation specialists in bioinformation to calculate the energy using a crystal structure data. What happened when you change the amino acid residue? Okay, so I failed to. I I included, but it's somehow missing here. But this is the data of the aspartic acid here. Okay, this is a side chain of carboxyl side group. Okay, carbon, oxygen, oxygen, and proton can interact here. And cysteine residue here is this is a sulfur, and proton is somewhere here. We cannot show the proton, too small. And then from the calculation, because the cysteine is smaller than aspartic acid, okay, as you can see here, this is larger, no? So new water molecule is fixed here. W is water in the presence of cysteine. So water is also can be involved in exchanging proton. So in this space, smaller space, but it's a proton. New proton is water is can be accommodated. But this is calculation, okay? Here and also here. In the presence of aspartic acid, it's difficult, but cysteine smaller in space, so two water molecules can be accommodated. So this also can be involved in also a proton of this hydrogen atom of this cysteine. So that's the interpretation. Why cysteine only not the serine? This not doesn't show serine, but serine from the calculation this structure is impossible. So, and energy level is energy must be higher to lower, so to flow the proton, but the serine is impossible. So we did amino acid replacement engineering and analyze the phenotype, PS2 activity, accumulation of PS. But still we need this calculation of molecular dynamic. It's a huge calculation. I don't know how to do that, but computer works 1 day or 2 days to calculate this. So then they nicely accommodated to water molecule here in the system mutant. So if you want to do engineering of protein, sometimes if you just want to know the activity change, it's okay. But if you want to know the mechanism, how this amino acid substitution affect the structure or function, activity, this kind of experiment. Well, of course, if you can make a structure analysis of this mutant, it's possible, but it's usually recently we can do cryo-electron microscopy, but it's very expensive. Takes time, and you have to wait a long queue to get access. So this calculation helps us understand the mechanism. So in future, maybe you may be involved to know the structure, protein engineering to develop a new, I don't know, medicine or new enzyme or medicines. This process must be done. Just only changing amino acid is not enough. Analysis of the activity in detail, molecular level, and structural analysis using a computer or cryo-electron microscopy is important. But this protein engineering is also very still need to get evidence how, why, what happened. So that's all. That's all. Any questions, comments?
So this structural crystal structure is already in the internet, right? Then we will analyze it.
Yeah, it's this calculation is you need a large computer, kind of super. I don't know they're using a super or cluster of computer, huge, or connection to the supercomputer. If you have a cluster in a one room, still takes 1 day or 2 days or 3 days, continuously calculating. I don't know how they do, but it's a lot of energy, electricity you use, and also mostly cooling the computer. So very energy consuming the system, but so you need a collaboration with specialists. Any other questions? This is almost ending, so final. Okay.
Any questions?
I think it's just what I want to show is not detail of the data, but the process of the approach, how to tackle to resolve the questions. So you need, as you can understand, we need interdisciplinary collaboration to do this kind of work. No, we can make a mutant, but we can analyze only basic activity, but detailed specified activity or structure analysis, we need to collaborate with the specialist of the structure biologist and the computer, very specific in the software. So that is, maybe you need to think about this collaboration. It's important. So human interaction is also very important for scientists. Okay, thank you very much for.
Yeah. So
Nice.
I think now it is all the theory. You can say the theory classes or theory courses are over now, and we have a lab today. And we hope you have enjoyed all the teaching, whatever teaching. 24 hours teaching is mandated by the GIAN program. So we have fulfilled all the teaching, and I hope you understand something, or you might have grabbed something. But if you have any queries, you can write to him or write to me or whoever the speaker. You can take the email, you can write to them and get clarified. So since you guys are MSc students, so we have a very good diplomatic relation with the Japan, right? You know that diplomatic relation is a good connection. Our government, Indian government and Japanese government, they are good friends. And there are a lot of fellowships for our PhD students who want to do for PhD or even masters. There are a lot of programs offered by Japanese government. You can apply for it. If you want to do it, please. That will be a very useful. Maybe you can send him an email and you can connect some laboratories. All of you are master students, right? Yeah. It's a good opportunity for all of you. There is a Mombush fellowship. Mombush? Yeah.
Ministry of I don't know in English. It's long.
Yeah.
Long name.
So that is also, there is a fellowship. It's specifically given for PhD. And also now started Lotus is the other program for people who want to do PhD. That is the other one. There are a couple of them. There are a couple of the fellowships. Otherwise, you can also contact directly professor, where you can be a potential PhD candidate in Japan. And of course, postdocs also, there are several postdocs. So hope we have enjoyed, and now we have one more day. So tomorrow, after the lab, tomorrow we have a quiz program that is also mandated by the GIAN program. All of you have to be there for a quiz and enjoy. It's not that we are testing your knowledge. We wanted to understand how you have grabbed how much knowledge you have gained. They just want to test and think that Venkat will be organizing that, and now of course we will be there. And then following the validity session around 12 o'clock. We'll finish it up by 1 o'clock or even early. So there will be some presents. Those who answer well. Presents. I think money is planned for some presents. So you have to prepare maybe the notes we have, whatever notes we have written. You go through, and only it comes from only the whatever Mr. Takashi and myself, Patika and other, whoever I thought, from that only we will get the question. Now we wanted to know that also with this feedback also we have to send to the NHR. GIAN is operated through NHR, Ministry of Human Resources. And they again we said the feedback, you have to give feedback. I also give feedback on you, how you have answered, that he will be telling based on tomorrow, and he will be also giving a feedback. So these are all cumulatively, they will develop program to the next time. So with this, if you require any help, you can also contact me. University of Hyderabad also is very well good institution in terms of research and teaching. So those who are MSc students, you can also join a PhD program, department of plant, any department. So I again thank you very much for accepting our invitation.
It was my
and
pleasure.
Hope we had a very good time with.
Already we had a very good time here.
So thank you. Then tomorrow we'll talk a little bit more. So okay, let's go for lunch. We'll break.
Okay.
5 people will like.
Come. Maybe you can also. Okay.
Okay, I'm not blocking.
So anybody did HPLC already? Have some idea about the instruments? So I will give first a general introduction about the parts. What are the parts in HPLC? Then I will talk about mobile phase. You must have know the theory and all, right? So like mobile phase, stationary phase, these all theoretical part you know. Then I will explain a bit about that. Then we will try to run one sample. First we will see the blank. There is some problem now with the detection. We are not getting a correct baseline for this, but we will try. If it is coming, we will also inject the thylakoid. We isolated, no? I kept it for extraction of pigments in acetone, 100% as acetone. Then we can take the extract and we can inject and see whether we are getting chlorophyll, carotenoids, everything in peak. So in HPLC you have a detector part. This is the detector part, and this is like the pumps which is going to carry the mobile phase and all through the column. And this is the pump 1 and pump 2. Now to open the pump and regulate, you have 2 knobs here. Then what else? So here this is like a reverse phase. What is reverse phase HPLC? Theoretically, depending on the polar phase of stationary phase and mobile phase, you can tell. If the stationary phase is non-polar, you call it a reverse phase HPLC. If it's a polar, then you call it a normal phase HPLC. Now, and what is HPLC? High
Pressure.
Yeah, it's two-way, like high pressure sorry. Hello. So what high pressure, in older times it was more focused on the pressure of liquid chromatography, because you are going to pass through a very narrow column, which is like very small pore size will be there, and when you pass something through this, and when it is going to a very narrow down micrometer pores, it will be in a high pressure, right? So that's why it was called high pressure liquid chromatography. But now it is more focused on its efficiency and performance. Nowadays people are calling it high performance liquid chromatography, and the pressure and pore size is also now more efficient. Like more narrow down, you will get the more separation of even minute compounds, whatever and all, it will be more separation, right? And what is the basic principle for this? On what basis you are going to detect the compounds? If you take thylakoid or whatever extraction, pigment extraction, you are going to extract many kind of pigments. Carotenoids only, xanthophyll, beta carotene, alpha carotene, all this you are going to separate. Chlorophyll A, chlorophyll B. So what is the basis you are going to get? What is the major principle you are going to separate this? Anybody remembering the
Partitioning of the compounds, partitioning between the mobile phase and the stationary phase.
Yeah, that is one reason, like the polarity you can tell. If it's non-polar, whatever going to interact non-polar with the stationary, because stationary is non-polar. Okay, so the which compound is more non-polar, they will interact with the non-polar things inside more, and which is less non-polar, or they will move faster. This is one reason. But even if it is non-polar, everything, what basically changes in your detection? The separation time will be different. Everything you are going to inject through here as a same compound. But once it reaches here, some things will be stuck in the first, some things are going to flow very fast, which is like not much interacting with the stationary phase, but they will be more, whatever the solvent we are giving, they will be more soluble in that. So when mobile phase passes, they will go with the mobile phase. Retention. So they are retaining there. Whatever is getting retained, some are not retaining, they are going faster. So the time of separation is different, and this time of separation is called retention time. This is the major principle of HPLC. And depending on this retention time, we will get, so when we normally do the pigments here, when we extract the whole pigments from algae and when we run, we get like the carotenoid peaks you will get in a 3 minutes, 4 minutes, 5 minutes, depending on again the mobile phase you are using also, right? It is going to interact with both stationary phase as well as it might be more soluble in your mobile phase also. So we normally use acetonitrile, water, methanol mixture. So in this mixture, and also there will be particular ratio according to what pigments you are going to separate also. These all you can standardize. If you want to more focus on beta carotene, then you have to standardize something you are getting beta carotene, the maximum peak, maximum separation and all. This you can play around. Okay. Now 3-4 minutes we get carotenoids, but we get the chlorophyll peaks after 10 minutes only. So this is the retention time difference. Now again, so separation is done. Once separation is done, where it is going now? Here. So what is here? This is the detector part. So once it is separated, now we are going to identify them. Identify, as well as we're going to do 3 things in HPLC. Separation, then this part is going to see the detection, like what is it inside, is it chlorophyll A, chlorophyll B, whatever it is, and it is going to quantify how much is there, what is there, how much is there, and separation. 3 things is going to be done here. And what is here in detector? What will be the detector basically? What can detect something with a wavelength? It's a UV visible spectrometry, right? It's a UV visible, normal, how a UV visible spectrophotometer works. Here there is a flow cell in which things will be, first carotenoid will come, then chlorophyll A will come, then chlorophyll B will come, like that it will come and it will go out through the outlet using this pump. So everything will go down. But there are HPLC which is connected to the, like you can collect it pure. From here the flow cell, if carotenoid you need, there are advanced HPLC techniques where you can collect the pigments also separately. That is also available, but in this case we don't have that function. It will go as a outlet, that's it. So here normal UV visible spectrophotometer will be there. So each pigment has a particular wavelength. So already according to the retention time you already separated it, you know. But to avoid more noise and background signals, when you are extracting the data, you can give the specific wavelength of each one, so that you are double checking the things. According to retention time, you know, okay, 12th minute or 12.5 minute you get the chlorophyll A peak depending on the mobile phase. And also the major part is you will have standards. Before running any crude sample, you will definitely have standards for what pigment you are going to focus on. Whether it's bacteria chlorophyll A, normal chlorophyll A, plant chlorophyll B, beta carotene, xanthophyll, violax, and everything, commercial standards are available, the purest form. So before, what you will do, you will prepare your mobile solvent, whatever it is, and you will run the pure. So you know it is chlorophyll A. And here, depending on the same mobile phase and stationary phase, you will get a peak. For example, chlorophyll A, I get like 18th minute, I got. Now I will run my crude sample. If chlorophyll A is there, you will have a peak at 18th minute also. This is how you get retention time, identification of chlorophyll or pigments, whatever. I am only talking about pigments, but you can do any other sugar, amino acids, whatever organic compounds, anything. If you have a proper knowledge about the mobile solvent to keep, and also there are different kinds of columns you can keep. This is C18 column. Carotenoid better we use at C30 column. So again you have to standardize which column you should use for your compound. That is also very important. Then C30. C30 column we use here. So we normally use C18 will separate everything, whatever. So we use C18, but it will be more specific if you want to separate different kinds of carotenoids. This is more specific C30. If you are more focusing on only carotenoids, you can use C30. It will separate each carotenes in a better way, C30. So what is C18, C30, what is this 30, 18 and all? Number of carbon. So this this stationary phase is basically silica, and in the silica surface, carbon chains, long carbon chains will be in this surface attached. This is called the mobile phase, and in this case, a C18 chain will be attached. In C30, it will be carbon 30. That's it. And polarity also I mentioned how polarity works, and here it is non-polar in a reverse phase. So you have any doubt or any part you want to know? Just this is the column, this goes here, here you are going to inject, this will take the whatever things you are taking, here it takes the things, and again this connected to the flow cell now through the white, and from flow cell detection happens, and from here the outlet you can see here. So this will take the whatever you are running, and always you will be having a stream of mobile phase. From here everything goes to here through the mobile phase. That's it. So these are the general parts you should know when you are working with HPLC, but I also don't know more technical part of this. This much I know, and if you have any doubt in this, I can.
Pump speed.
Pump speed, again the flow rate. Flow rate now we are keeping like a 0.4 ml per minute. But this also matters when it comes to the retention time and all. You can standardize this in which flow rate it is more good for your compound. For now pigments we keep a 0.4 ml per minute, and yeah, that's it. Yeah.
The mobile phases, what is it? Methanol, what are they?
Acetonitrile, methanol, and acetone. 70, 20, no, 20, 10. This ratio we are using.
70 is acetonitrile.
Acetonitrile 70, then
20 is acetonitrile.
I forgot that. I will check in my book. I have doubt. I forgot that. I'll check that one. So this again you can see papers where they do, and you can choose, and you can use maybe sometime switching the mobile solvents. Once the carotenoid done, you can switch to pigments like chlorophyll. Then you can change the mobile phase. Maybe we know methanol is more good for separating the carotenoids than acetone. So in the first half, in 4-5 minutes you are getting carotenoids. You use a more methanol saturated mobile phase. In next 10 minutes maybe your chlorophyll going to separate. So then you can switch to the other mobile phase. So if you are using only one, it is isocratic. Otherwise it will be binary, like 2 you will use. Now let's try to
Run and show.
Yes. So this is the software lab solutions we have. Okay, wait. First I will show. It's not working. Okay. So we'll switch on all the parts. Detector, then pump 1 and pump 2. And first we use a purging. Purging means instead of that flow rate and all, you are just going to like flash first mobile phase. If any air or something is there, it will go. So you ask now the pressure. So we keep, now it is 0.3 ml. And in 0.3 ml down, how much pressure is there? What does it mean if the pressure is too high? If the pressure is too high, what's your understanding?
Retention time will be affected, I think.
No, I am just not injecting anything, nothing. Just the mobile phase I am going to run. And then also you are having a very high pressure. What does that mean? Something is stuck here. That's why your solvent is not moving in a normal pressure. Something is stuck there, the pore size is again clogged, so the mobile phase is not able to flow properly. That's why you are detecting a high pressure here. So what you should do? You should clean it. You can clean it using 80% methanol. You can also clean it using very warm, like little bit hot, 60 degree, like that hot water you can use to wash the column. Even isopropanol 100% you can use to wash the column. This all will wash out any compounds get stuck inside the column, it will wash out. You can do it for a day like that, it will wash. Now I'll do just purging. Whatever this inlets and outlet tubes, if there is something stuck, it will just, now it is not going through the column, it is just cleaning the outlet inlet pumps. For that you are using very high speed, there is no flow rate, nothing we gave. It's not going to detect anything. So the purging is going on. This takes 1 or 2 minutes.
To use the solvents of HPLC grade only?
Yes, yes. And where is our mobile phase? We don't have? Acetonitrile acetone. This is methanol, right?
Yes. I kept here only.
Not this. You prepared both, right? Okay. Yes.
70, 30, 20, 10.
Methanol, acetonitrile, acetone, 70, 20, 10.
Methanol is 70.
Yes, yes.
Acetonitrile
Is 20.
Acetonitrile.
And acetone 10. This all will vary again what you are going to work with. These are all just what we are using for now.
No, but this is common for pigment.
This is common for pigments. Yes, yes. Chlorophyll, carotenoids, everything we do with this only. And we don't change, we use isocratic only. If you are more focusing on very specific beta, alpha, you want to separate the carotenes in that way, then you can switch to some other mobile phase, also stationary phase.
Sample is ready.
But I am thinking sample I will show only one time to every people. It's just injecting.
Yes, yes.
I will take the other people and explain the parts. Then we can go for an injecting.
Can I keep the sample inside? Because you kept for 10 minutes. I'll keep on rotating.
Centrifugation is done.
Yes.
Directly we'll bring here.
That's what I
And I am thinking one time to filter that one.
want me to filter.
Is there enough? Or add little bit acetone and you can filter it. You take this whatever supernatant, don't touch the pellet. still green is there in the pellet?
I'll just see. I didn't see it.
Then if it is too less, add extra. Show me once.
Okay.
So now purging is done. This is the window. You will see the fluorite and everything. Once it is done, these outlets things you should close. Now detector is on. Detector was blinking, right? Once it is stable, you can open the software. So we are using this lab solution. And also this mobile phase, whatever you are using or the cleaning washing solution, you need to degas. If any bubble, air bubbles, and when you prepare mixing and all, there will be a lot of gas inside that. So you need to remove this, otherwise this gas going to stuck in your column and everything. So then it's gone, you can't use that. So you need to degas it. Either sonication you can do, water, that backside you will see sonication, it will degas. Otherwise there is vacuum infiltration method also. Yeah. Yes, it will.
Then you'll explain the next group. You'll explain.
I think I will. I'll show this people software once. And HPLC you go. These all know you will learn once you're going to do the sample your own. If you just, even in our lab, whoever continuously uses, they will have a experience, they will understand, okay, what is the problem and all. So practice is only these kind of instruments you can learn. For now you can get some idea, and when you go to a new lab you won't be like, oh, what is this instrument, I don't know how to operate it. One person is explaining you will be more able to understand the things. That is the best part now you are getting. But don't think that by this one time you are able to do that. These are very advanced, so it will be very sensitive also. If you do some mistake, again this software will stop once you do one error only. So should be very careful. So this is LC mode, which is like it's local mode, right? There is no detection, nothing. But if you want to see the detector, that is PDA, which is the detector part, you will see here in how the running is going on. This is the window, you will see the peaks and everything what is coming, and again it shows the milliampere and which wavelength now the pigments are getting absorbed. These all will come here, and this is a representation of the column. If some pigment is coming here, it will show in which wavelength that color here it will come. So you are seeing, okay, there is something is coming. So this is the detector mode phase you want to see when you are running something. For now we will just run a blank, and this is the one to inject and load. So this is like injection mode, and this is the loading. Once you load, you can inject it. Loading should be here. Once you are injecting, you take it down, this one. So it automatically takes the things. Now I just want to see the mobile phase which is coming a straight line. So what I will do, I don't have to inject because already in the stream it is there. We just need a detection. So I can down, so it will take the detection part. Let me explain the other people so that I will show this that time. Okay? Any doubt or it's clear? Okay. Remaining people can come together now. How many are there? ഇന്നലത്തെ ഞങ്ങൾ ഫുള്ള് റൺ ചെയ്തില്ല, പിന്നെ 5 hours ഇല്ല. I will explain you with some. CD, yeah, CD one of us will show, maybe Jyoti. Okay, I just opened everything. I will try. So anybody done already HPLC? You must have used. You also did. Okay, so whoever don't have any idea about the instrument, I'll give a general introduction. If you ask me too much technical insight, even I also don't know, but I know what is the functioning and which part functions for what and all. I will explain you. So what is HPLC? High pressure liquid chromatography. You can call it high pressure. All the time it was calling high pressure. Now it is more not about the pressure. Now it's about the performance. They are improving the performance by narrow down the pores and everything. Whatever available earlier, now the pore size and all very small. So you can separate whatever things possible, right? Like some pigments, alpha and beta, which are very close structures. If you want to detect them, there should be that much narrow down pore size to separate this both. So now these are also available. So now more it is called high performance, but it is always working under high pressure because you are injecting something and allowing them to go through a very narrow. Here it is a pump, they can go easily, but once it enters in the column, there are minute pores which has to go, which will automatically generate a high pressure. Okay. Now this is the detector part. This is pump 1 and pump 2. These two are taking the inlet and outlet of mobile phase, and in so pump 1, pump 2. So now coming to the detector, what will be inside the detector? Yeah, what is going to detect?
Screen.
Maybe a little bit more, because when it passes through the filter you won't get anything. Understand?
Acetonol
Add little bit,
100 microliters.
maybe 250.
Can you give the filter?
Yes. So in detector part, basically there will be a UV visible spectrometer. There will be a flow cell. The things I inject in this part, you can load here and take the knob down. It is going to be injected, and from here through this it takes to the column. From column it is going to separate depending on affinity, basically affinity. So how you tell this affinity? There will be polar and non-polar compounds. As we have a reverse phase, which is like non-polar component will be the stationary, and you will use a polar phase for the mobile. And here whatever I am injecting, something is non-polar which will more interact with the non-polar material inside. They will retain more, and which is less non-polar or more polar, they will go with the mobile phase. They are more soluble in the mobile phase. So the things get stuck, which is like non-polar, or you can say hydrophobic. Hydrophobicity will be more. They get retained, or they go very slowly. They are not ready to move. So they go very slowly, and which whatever goes with the mobile phase will be take like this to the this part. Here there is a flow cell. Once one by one things will come. Here it's already separated. First some keratin will come, then there are several different carotenoids. So one by one they comes to the flow cell. Once it reaches the flow cell, the spectrophotometer will work, and depending on the wavelength and all, here the detection happens and you get a peak here. It will be amplified detection, and it will be amplified as a peak here, and the peak represent the quantity or the amount of or the concentration, you can tell. Okay, so three things happened here. After injection, separation of complexes happened. Then after separation, it's going to be identified what is what, and once identification is done, the third thing is quantification. How much peak you are getting, that peak area corresponding to the quantity, how much nanogram, how much microgram or nanograms are detected. This is going to be three things. Now I explain how you are injecting, and also you people know it depends on the affinity. So how identification is happening? One way is wavelength, but before that in column how it is getting separated, or
More size, more polar molecules will move.
because of that what is getting affected? The time of separation is affected, right? time of separation is affected, which is called retention time. The retaining, at which time they are getting separated. This is called retention time. So chlorophyll A molecule will have one retention time, beta carotene will have another, xanthophyll will have another, lutein will have another. As these all carotenoids have similar molecular structure and everything, they will be separated in a very narrow range. Maybe 3 minutes to, normally depending on the mobile phase we use, we get like from 3 minutes to 6 minutes it gets separated. All the carotenoids get separated. Then it is not going to show, okay, this is beta carotene, this is xanthophyll, nothing. Then how you will know? It doesn't know this is beta, this is nothing. So how you are going to understand? Okay, I got a peak in 3 minutes, also a peak in 4 minutes and 5 minutes. How I am going to detect which is carotene, which is xanthophyll? How I am going to detect it? But all the carotenoids will have a range like 450 to whatever, 500, 550. These range they will. So you can't, even all this range they will absorb. It's not like, and even chlorophyll molecule can 400 to 700, they can absorb any range, right? So then how you will understand? Compared with, yes, not already sample. You are going to compare this with a standard sample, which is like the pure foam you will get. It is commercially available now. You can buy chlorophyll A, and this will have a known concentration also, like 5 microgram or 5 micromolar, whatever concentration they are giving. You will have a known concentration, known molecule. So before you are going to run your sample, you need to run a standard. Once you run the standard, there is nothing else, only chlorophyll A. So you are going to get a single peak, right? And that single peak will be either 16 minutes, 12 minutes, whatever. You are going to record it. So now you know chlorophyll A, if I use this specific mobile phase with this specific stationary phase, I am going to get a peak in 16.5 minutes. You know now. Now you take your sample, algal sample, you extracted the pigment, everything is done. Now you inject. You will get many peaks, but something you will get in the 16.5 minutes. So the retention time is the basis. With a standard you will get. And comparing this peak with the known peak, you know one known concentration and unknown concentration, you know how to get that. Known and unknown concentration, you know the calculation, right? Known concentration and unknown concentration. Now you are going to get the area. So you identified and quantified. Understand? Then what we missed? Okay, what is the stationary phase here? It's silica material, and silica material alone you can't use. You are going to know the polarity and all. You need for more efficient separation and everything. So you will have long carbon chains attached to the surface of silica molecule. This makes it non-polar, polar or non-polar. If it's a normal phase, it will be polar. If it's a reverse phase, it will be non-polar stationary phase. Here we have C18 reverse phase. So it is C18 attached to the silica. And mobile phase, for now we standardized, we have like a ratio of 70, 20, 10, methanol, acetonitrile, acetone. This is for only for pigments what we are working. If you are focusing on chlorophyll to carotenoids, everything we separate using this. But someone is more focusing on carotenoids, you can change the mobile phase. We need a more general approach, that's why we are using something mixed with acetonitrile, methanol, acetone. If you are going to focus more on carotenoids, you acetonitrile, methanol, acetonitrile, acetone, 70, 20, 10. But you refer to more papers and focus on your part. You might not be needing chlorophylls. You may be focusing on carotenoids. You can go for those papers which focuses on carotenoids. You don't have to follow this.
This is reverse phase.
Yes.
In normal there will be no carbon chain.
It is there, but it will be like the group functional groups will be different and it will be polar phase. it won't be non-polar. This is the difference between reverse and normal phase. Then what else we missed? Everything is explained, I think.
This pump one, what it does? Pump one also gives it for injection, or only pump two gives for injection?
Both. When you use it for like you want to use two phases. Okay. Both do the same function. Inlet and outlet is connected to both. Yeah. And I was talking about isocratic and biphasic. Like if you want to switch, in first 5 minutes I get all the carotenoids, and next I am going to expecting the chlorophylls. What should I do? Which is more good mobile solvent for carotenoids I can use in first phase, and second I can switch. You can just program it and put two pumps separately and it will change. That's it.
The size. What is the size?
Which
No, size. The diameter.
no, I don't know exactly. I don't know. C18 column, if you type, you can get that one. I don't remember. They don't mention here, I think. Huh?
Software.
This is lab solution. Any doubt?
More than C0. Japanese company.
Now we can. Okay, I just opened lab solution software. You get a window like this, and first I'll explain this monitor also. So here you can see something ml, 0.3 ml, which is the flow rate. Yesterday I told no, this will regulate the flow of whatever is coming from. So in the same way, here 0.3 ml per minute is going on in both. These also matters, the retention time and all, how much you want to inject. If you inject in a very slow rate, it's going to detect like very slowly, and the separation will be like very slowly. You will have more separation or something. This also you can standardize. Then this is something Pascal, which is like the pressure inside the column you will see here. So I am not running any compound. It is, I am just going to run now blank. The pressure should be not zero. Some solvent is going through inside that. Zero means nothing is going. Something is going, there will be some pressure. Blank, you should get a minimum pressure. But I am getting like very 40 MPa or something I am getting. What does that mean? I didn't run anything. I am not injecting anything. But still I am getting very high pressure. Why?
It might not have been washed properly.
Yes. Previously something is inside. That's why it is getting stuck, and you are getting, something is hindering. That's why you are getting a pressure there. Okay.
This is silica. Inside is silica.
Don't touch. Yes, inside it is silica.
Oh, it is normal stainless steel.
This I don't know. It should be. So I opened it, and this is the local mode, which is like only about the what is happening here. But you go to PDA, which is like detector mode. It's going to the detection part, and you get here the different peaks, and this is the time of peak. Like you see minute here, you're going to detect here, and this is all about the wavelength and what voltage it is going on, milliampere, and which wavelength what is coming here. You will see some peak is coming here also. It will show, okay, this wavelength is absorbing it and all. And here you will see a representation of the column, like you are getting something in this wavelength red, like this wavelength you are getting something blue color. So you will see a blue stain here, something is here, you will see here. So this is the detection. Now the LC mode is also ready, and also the detector part is also ready. Now before injecting some sample, we will see blank, because we want to see any like earlier some things are inside, Otherwise, whatever we are getting peak will be from the previous also. So you need to do a blank for the reference. So maybe you people can also come. I will on the blank for some time and we will see whether we are getting a straight line filtered. Keep in there, we will keep 10 minutes blank, we will see. Enough, 5 microliter we will inject. Okay, I will open some method file. Okay. Okay, these are all some method files, like how much time you want to run, how much pressure you want to, how much flow rate you want to keep. These all you can program. Then you can save it as a file, and I am just opening that file so that it's set for, see now it is showing, yeah, I am just downloading and closing it. Here you see the LC stop time is 10. We will just keep for maybe 20 minutes. And here you can keep, I am not keeping any time program, and pump flow rate will be 0.4 ml per minute. This you can change if you want, but I will keep it 0.4 for now. And it's an isocratic flow because I am not going to use any two-phase and all. Then this is the detector mode. If you want to change something, now the wavelength is from 190 to 800 nanometer. And controller, you are not going to do anything with this. And yeah, 0.4 ml I did. Then what? This is all.
That 20 minutes is for?
So that it will detect for the 20 minutes and it will stop the program. So let's keep it 20 for now. Okay. Now download and close. And now, so if I want to, now I want to inject my blank, but it's already in blank and the column is already streaming with the blank. I don't have to inject again blank, I just have to give an inject mode, right? Shit, someone did that. Let's see. No.
This.
Mobile page itself is a blank.
Yes. Somebody already did that.
Mobile page is blank. Same.
Mobile face is, for now I am keeping it as a blank. Yes, mobile face itself will be the blank. Yeah. No. Nothing. It was like in this mode already, so it's now not getting connected. But yeah, it will be after some time. It will get connected. For now, it won't. I'll try one more time. No, should be only one beep, then only it will get connected. So we will wait for some time and then only it will get connected. See, it's not getting connected. I'll switch off and on. Maybe during this time you people can do CD once and then you can come back. After this you can come back and by that time it will be ready for connecting. Then I will show one sample. Before that I'll do one baseline.
BNG page?
BN page after all this I will show. No, it was not down. It was upper.
Whole time.
Huh. Then it's not getting connected. I switched off everything once. Jyoti is not here. Can you show one CD?
Yeah, no. If you can also go for secondary structure of this. It will show your what is the amount of alpha helix, what is the amount of beta helix structure in a sample. It will show you things. So the wavelength will change. Wavelength will be here. It is from 400 to 800. Here the wavelength is from 400 to 800. That wavelength maybe it's from 190 or 180 to 200 or 250. That wavelength for secondary structure of proteins. Okay. So
sample analysis.
I forgot to bring the pipette. Can anyone bring pipette and tips? 1 ml. 1 ml pipette and tip.
For this analysis, for sample, how much they will charge? So, suppose in outsourcing, if you
I don't know about the charging thing, but chemistry people come and use it. Maybe Dashrath knows, the lab attendant here. Maybe he will be knowing.
But in outsourcing also they will do. No?
Yeah, you can do.
Chargeable.
Yes, it's with chargeable. Yes. So first we will load our. This is your STN2. Okay. So because we already dissolved STN2 here, not dissolved exactly, suspended. So dissolved will be in acetone. This is suspension. So some people, if they centrifuge it, it will be completely white. So all your thalaggoids will be settled because this is not acetone. Acetone will dissolve your pigments. Okay, all organic solvents will dissolve your pigments. This is not organic solvent. So that's why you should not centrifuge it. You need to vortex it to equalize all the suspension. Throughout the suspension should be equal. That's why you should once vortex. I did that time, right? Vortex. So after vortex you can go for reading. And another thing is that it should be protected from direct light source. This much light is present, that is fine. But you should not show it directly to this light. Got it? Because these are all chlorophyll pigments. If it is protein, then that is fine. Pure proteins, that is fine. Since it is chlorophyll and chlorophyll is having also pigments and proteins, you should not directly show in light. Another thing is that it will run at room temperature, but before that, for the stability of the sample, you should keep it in 4 degrees.
But there is that yesterday we used like liquid nitrogen. Why we are not using here?
No, liquid nitrogen, this is the work of this instrument is different. That is fluorescence. Okay, this is circular dichroism. This is a reading, it's not a kind of any fluorescence kind of thing. So it does not rely on your like excitation of a molecule and then recording the fluorescence. It will direct see the chirality of a molecule. Okay, so the working may be same, but the entire protocol, the working principle means what can I say about this thing? Like that one, you pour liquid nitrogen to freeze your sample. Because normally the fluorescence, 95 to 97% of the fluorescence at room temperature is from PS2. That's why you are getting a BYFM in room temperature, everything. So it's not like that PS1 is not having fluorescence. PS1 is also having fluorescence. But that fluorescence is very minimal and that is overlapped by your PS2 fluorescence. That is the reason you can't detect PS1 fluorescence at room temperature. So you need to first stabilize all the molecules, means completely freeze it. Once the molecules are freezed, the excitonic interactions and the vibrational interactions will be completely halted that time. Once it is halted, when you excite it, so 2 peaks will be seen properly. In higher plants, 2 peaks very proper to, but in plants, complete 2 peaks will be seen. So 2 ml we will add, right?
Yes.
This is also quartz. It's clear now, you will not be knowing which is. Yeah, 2 ml is more than sufficient.
It's blank, sir.
Yes, blank. Just so make sure whichever number you are putting, whilst you are putting, I kept in 3, right? So same 77K, first you have to run blank.
right? So acquisition, I kept only one acquisition, not 3 acquisition. So just go to blank. So specified cell, this is important. So cell I kept in which cell number? 3. So if you place here 1, it will not take any readings. It will take readings, but you will not get any results. So you have to place a 3. Or if you kept everything,
All cells.
you can click all cells. Okay. So anybody has that protocol book? Achhi book. Nah, then not sure. This is for molecule. Okay, fine. So while running this thing, you see this is control. This is a 1, 2, 3, 4, 5, 6. So total 6 cells are there inside. So now we specified cell number 3. That's why this one is highlighted. The temperature besides it is written 24.82. So if you really want to run your sample at room temperature, it should not exceed 25, right? So what happens is that if you run for too long, 2 hours, 3 hours, the temperature might go to 26, 27. So that time you might get faulty readings. Got it? So you should be careful about these things. That's why this temperature is shown here. The holder temperature and each and individual cell's temperature. This is the average, I guess. So this is the individual cell's temperature. That is important. So this cell number 3 temperature is 24.7. As far as like lower than 26, that is fine. Until and unless you want to exceed the temperature to 27, 30, 35, it can go higher also. 100 degrees it can go.
How long?
Entire day. Entire day it will take. Yes, only baseline. You stay here, tomorrow you'll do the sample. No, it's not like that. See, baseline does not need to be 0. This is not your complete water or something, right? This is STN. Sucrose is there, triazine is there. So some particles are there. So it will not never be 0. Baseline does not mean that it will be from 0.
After finishing, this should be
Yeah, it will be 0. Only because only one accumulation I took. Okay, that's why. So another thing is that people get also confused about this baseline thing. So baseline means they will always think that it should be exactly on 0, right? It should not be like that. Until and unless you are using a kind of like, for example, distilled water, that is fine. But if you are not using distilled water, here STN2 is used. So it will have some amount of absorption and this chirality and this thing. That's why this thing, it will be negated from your peaks, whatever peaks you will get. Initially it will start from 800. It will drop to 400. Everywhere it starts from higher to lower. Why higher? From lower energy it will start to higher energy. Okay, higher wavelength means lower energy. It will ramp up its energy slowly. That way it is more efficient. If it will first ramp up the energy, it will consume energy. Again from ramping up, it will come to ramp down, it will again consume energy. So it's better to go from lower ramping up to higher ramping up one time only. So that way it is more efficient.
Can we put both the blank and sample together?
You can keep, but again you have to go for all cells. There is no need of that. Got it? So if you want to keep all 3, we only have 3 samples. So all cells you have to wait. Yes. You can't open it and take it out also. Once everything is finished only, you have to take it out. So unnecessary you have to wait for like 4 more blanks. Yes, done. Now we will put our samples. Same, this is nothing. Almost so why this thing turned this side, you know?
Pass through the monochrome.
Yes, light is coming from here. See here, the lens is there, na? Yes, that is the reason. It's a bit difficult to remove also. So these wires you are seeing, na? These are temperature sensors. These small wires. These are temperature sensors in each cells. So without enter 2 ml you need to transfer, so I am not taking it anyway. No, not an issue. This is not taking absorbance. This is different. This is basically the chirality and non-chirality. According to the rotation of the light, it will take your observations. So now you just run your sample. Okay. So cell 3 already. So it will now not rotate. It's already in that position, and I kept in that. You see, 2 major peaks will come, this region and this region. There will be both positive peaks as well as negative peaks in your thylakoid samples. Got it? So it's not always positive. Depends on chirality. Depends on the angle in which the light is bounced.
Where is the option run?
Here it was there, now it is not highlighted since I already ran it. Yeah, around this region one peak you should get, but I don't know, maybe the concentration, no. Yeah, it is rising. So this is only specific to plant sample. Algal sample will not be exactly same. Okay. Depending on your samples also, this will vary. But usually, if thylakoid, the thylakoid is from higher plants, you should observe this kind of. And also, if you feel like this peak is less, I use 20. But normally, for my work, I use 30 microgram. Got it? So I will, means like you can increase the concentration also. So another peak should come around here. Which one? Okay, this is voltage difference. You don't need to check this. This, because of this voltage difference, these peaks are coming. Yes, first one only you need. MDEG, that is the concentration of CD. That is in milli degrees. That is because it will rotate the light that way, right? either left or right. So depending on the chirality, this name is given MDG. Okay, this peak came around here. So this is the characteristic feature of plant thylakoid. Yes, carotenoid also. Not only carotenoids, this is actually all macro domain groups. It's not only carotenoids. It is a clumping of all your pigments and protein complexes.
The plant is under stress.
Yeah, usually this will drop. The peaks will drop. Yes, intensity will drop. Yeah, position should remain same. But in 77K, the peak shifting, sometimes it will happen. Actually depends on stress also. Sometimes here also peak. I haven't seen peak shifting in CT, but in 77K, under stress, sometimes the peak shifting is there in PS2. Okay. Once it is done, again we will go to, I think, control. Where is that? No, not here. That analysis thing I'm forgetting. No, baseline is done. Which one? This is no. Actually, the changes are based on the voltage. Okay. So this voltage change is detected as a peak. We don't need to take this one. Only this is necessary.
Then you have to plot separately, sir.
Yes. I think I forgot a long time back. I didn't. So again you have to go to send to analysis. So many times I clicked, that's why it's came. Okay. So just like that, I already told you in 77K analysis you have to click, go to processing, corrections, smoothing. Smoothing is not required, but you need to adjust the baseline. So processing, corrections, baseline. So same thing, you need to just check everything is fine or not. Okay, so here you see the peak is prominent. Once the running is done, just press OK. Once it is done, you can only this graph will be exported, this point. So this is the voltage difference. So you need not consider this. This part is actually considered. Go to file, go to export, go to CSV files, and you can just select your whatever. कर लो change. Wait. कहाँ गया GIAN ये? So you can just give your one CSV file, save. So just to cross check, this is the main running program. So this is the program, Spectra Manager. Okay, this is the CSV. So it will be saved as a notepad format. You can, these things you see, it's starting from 800 and it's going till 400. So this point you can copy and paste it in Excel. Okay, exactly one point will be extracted. So with respect to each wavelength, the point will be there. So then you just plot in XY, just normal plotting you do, will get the values. Okay, anybody wants to put second one? Okay. I will take it out. It's difficult to.
Energy level.
It's not energy level, it's the degree of rotation. So either it will be positive or a negative. So depending on whatever you sample, whatever it has. So sample is not means like it's not homogeneous, right? It's heterogeneous sample. So many different photo systems are there. All other proteins are also there. Depending on that, it will show either a positive rotation or a negative rotation. तुम बीएससी कोड़थिला किए पढ़ुले त्रिविक्रम से पढ़ुले मु बीजेपी बैच 2015 आम बेले त्रिविक्रम तो पढ़े बहुत जन लोक रिटायर्ड हो रिटायर्ड हो ना सर पुष्पांजलि मैम भी आम बेले आसते जॉइन करते No. Which one negative? That depends on this point. What point exactly it's running? Now it's beyond 800. That's why it's not recording. Okay. So just go to anyone wants to process it.
Process means next person.
Go to analysis. Okay. Once you click here, this thing will come. So 2 times it's already open. So we go to processing, corrections, baseline. Smoothing is not required. If I do smoothing, this peak will decrease. That is the problem. If you have too much noise, then you maybe can try. Otherwise, the peaks will drop if I smoothen. I will show you once. So this is the smoothen one. If you want to smoothen it, just go to see the peak size is dropping. This will smoothen, this will come straight line, but the problem is that the peak will decrease. So if you are comparing the peaks, the problem will come. So it's better to not smoothen at all. So this is done. Go to yes, see here, baseline is done means this should come at 0. Go to file, go to export. CSV. Press 2. Done. Okay, third one, last one, anyone wants to load? Okay, that's fine for learning purpose. This is fine. So now if I keep it here in 5 instead of, or if I keep it at 1, just remember I kept it at 1. So now I have to, once I start the sample, it will ask. So now you have to go to cell number 1. So now you have to deselect it. Then it will turn that to
1.
yes. So now there the 1 will be highlighted, you see. And that's fine. 25, 26 is. So if you want to increase the concentration, you can increase to 30, 40. Don't go beyond 100. 100 is also too much. 30 to 50 range should be enough if you have a good concentration of chlorophyll. P sample.
ये जो हमें जानिले कि कोरिलेटी से मतलब लाइट को डायरेक्शन को बेंड करला कोन होला तार साइजटा
साइज नो डिपेंड कन्फर्मेशन माने तारो देखो गोटे हेटेरोजेनस मिक्सचर रे एक्स रो कंसंट्रेशन 10 अछि आ गोटे हेटेरोजेनस रे एक्स रो कंसंट्रेशन 5 अछि कार रोटेशन अधिक हबो 10 रो जार सिंपल कुछ तो
ये अगर हमें कोन आइडेंटिफाई करले ये जो ग्राफ रो
देखो these are all macro domains जहाँ सब जितने भी रहा for example all these like those chlorophyll molecules and also pigment systems. So pigment systems are not only pigments. Pigment or protein, both it consists of pigment and protein complex. So pigment will absorb some light absorption will be there, and also proteins also light absorption will be there. But depending on their concentration, they will deflect the light in a particular degrees. Okay, that is in milli-degrees. That's why MD is written. So that slight rotation is detected here. Either it will be positive rotation or a negative rotation. Or no, this is a normal recording. Control sample should have. In some case, if for a particular photosystem complex, actually it is already available in papers, which peaks represent what. Okay, so you can consider those papers and detect. Yes, you just superimpose all your image, all your data with treatment along with your control, so you will know what change is there. In some region the peaks might not change, the lower peaks might change. Okay, depends on what you are using and which stress you are giving to the samples. But this you can correlate with your BN page. CD and BN page is usually taken in a single set of figure, so people can correlate. Yeah, last sample. Okay, so it is done. It's already open. Down, down. It's open already. Processing. Corrections, baseline. Yes. अन्ना इसको निकाल रहा, मेरे हाथ से फिसल रहा है. So going to that side, na, it's difficult to take out. I'll clean and give. I forgot to bring the distilled water. I'll clean and give now only. I'll give. Done?
That's it.
Okay. Now you can close it. Anna,